but I'm just going to talk about some of the upcoming reports in the first contract of EU for algae, so the stakeholder platform run by DG Mare and Sinea. And there's this new contract, as Talia mentioned, starting this year for the next 2 years. So there'll be more to come, but I'm going to look backwards at the final reports that are coming out from this first contract. And the slot here is the title is Market Trends and Monitoring Frameworks for Bioremediation and Ocean Regeneration. I'm not going to go into too much detail, but it's just really looking at two reports of several which are coming out, just to give you a kind of snapshot of what to expect. So there are more by some of the EU algae partners coming out, but these are kind of the main 5 reports coming out from the ESPRO side. So we are looking at social aspects, the bioremediation potential of algae, product LCAs, the business case for bioremediation, and spatial planning as well. So monitoring is a very core element of all of these topics. Yeah, as we'll see. So I'm just going to talk about 2 today because I was mainly involved in these 2. So one is the business case for bioremediation, and the other one is on social aspects for the algae industry growth, and try to talk about monitoring in both cases. So first, if we look at the business case, so in this report that's coming out, we've identified 5 use cases. So the first being here, bottom right, the decontamination, so the uptake of contaminants such as heavy metals. Secondly, nutrient recycling, so that's nitrogen phosphates, potentially from agricultural runoff or from fish farming, for example. 3, just as a biomass production for human consumption, like food, cosmetics, etc. 4, for ecosystem restoration, so like a much more regenerative approach. And then finally, 5th option here, and perhaps most interestingly in the report, is the sea to soil transfer, where those nutrients are actually going back to the land, and this could also be a bioremediation measure, sorry, on land. And I won't spend too much time on it, but one example is biochar, so where seaweed is cultivated, and then the residues from the so the side streams from seaweed can be burnt through pyrolysis to make biochar, which is very good as a soil conditioner, also as a fertilizer or a biostimulant. And if you look at some of the carbon credit schemes that are already existing, mostly terrestrial schemes, biochar is about 93% of the carbon dioxide removal methods that actually deliver carbon storage or even sequestration. So the other ones are actually very small by proportion, so this is why biochar is of interest in this report. And partially because the consensus for marine-based carbon or blue carbon sequestration is not quite there yet. So we're looking at ways to kind of dock in or find existing credit schemes where algae, particularly seaweed aquaculture, can play a role in the short term. So the key takeaways from this report are that monitoring is crucial for both the licensing of new farms, whether it's on land or at sea, and also for credit schemes, in order to kind of validate, report and verify the carbon uptake or sequestration or storage. Secondly, monitoring tech was one of the fastest growing markets identified of the tertiary services, so that's technology and service provision. So this is monitoring devices, monitoring tools. There is a lot of potential there for innovation. High permanence carbon products, such as biochar, as we mentioned, bioplastics, bio-based materials, but also larger engineering solutions like nature-based solutions should be prioritized to be in line with the EU's certification, carbon removal certification framework, and also recommendations from the IPCC. And finally, that multiple quantifiable benefits are needed in order to balance costs. So in other words, to be regenerative, which is this term that is increasingly used. What does that actually mean? It means that we need a net benefit, basically. And this was a screenshot from a European Investment Bank report showing that you need these benefits to work in tandem. So the increased fish catch, the carbon sequestration, biodiversity, nutrient uptake, etc. So they all need to be quantified, monitored, and demonstrate a benefit in order to be kind of classified as regenerative, but there is no official definition of that yet. So then if we look at the second report, this one is about social aspects, so less about the ecological or environmental monitoring, but more about the socio-economic monitoring. And of course, we look at the just transition. So how does this work in the algae industry? And it's very much focused on redistribution of value and equality and fairness. So and things like, well, local economics versus macroeconomics. How do you redistribute value within a small community? How can small coastal communities play a role in the algae industry growth, both for micro and macroalgae, although the report focuses mostly on macroalgae. Yeah, and how should we kind of keep an eye on the growth of the industry to make sure that it's fair and it's actually keeping wealth within Europe and within communities. And there are schemes that are increasingly facilitating this just transition. There's one example here in Germany where fishers are being, there's a training available for them to do ecosystem restoration, such as kelp restoration. And then on the right here, it's the Sea Ranger Service, which is a Dutch initiative to get young people into marine surveying and monitoring at sea. So those are two examples in the report. And then we recommend a set of indicators actually based on this report in Nature Communications Journal, which is a very long list of recommended indicators for aquaculture of various kinds. So not just algae, but finfish, shellfish, and so on. And so we just look at the social indicators in this report, where you can compare things like the managerial returns. So this is the compensation that the higher levels in the company receive versus the lowest, whether it's a benefit for community services, for health services, or whether it's more of a burden, whether the company is locally owned, whether it employs local people, and whether there are career opportunities in that sector. So there's a comparison here, for example, with salmon and seaweed, that local ownership scores quite low because these are often quite large investment companies that invest in salmon farming, whereas seaweed is potentially more balanced, but it's obviously very early stage compared to salmon. So the key takeaways is that these socioeconomic indicators are crucial both for informing policy and for ensuring fairness for a just transition. And some of these indicators that we've mentioned, they already exist for aquaculture, and it's useful if these indicators are cross-compatible between sectors, so not just between aquaculture sectors, but also agriculture, for example, so we can see how this just transition is evolving over time and whether it's fair for everyone. And then one thing that came up more recently was whether the algae community should be incentivized to submit socioeconomic data anonymously, like an algae census, so we kind of have a better picture of the volumes of production, how many people are employed, what's the gender balance, how much people are getting paid, and this kind of thing. So this would be very useful data moving forward, but we only have scattered data so far. So these were just 2 reports that I've talked about, but there are 3 more, and also I think even more than that. And the first one is on the bioremediation potential, summarizing results from ongoing or finished or ongoing projects. Secondly, on spatial planning and water landscaping maps or MSP tools. And finally, on product lifecycle assessment, so possible criteria labels and tools for that. So keep an eye out for these ones. That's it from me. Thank you very much, and we can move on to the next presentation.
Amazing. Good time management, Frederick. Thank you. We're going to have a question discussion session at the end of all of the presentations. So if you have any questions, please post them in the chat or keep them, and then we'll try to go through it all in a little bit. And with that, I hand over to Barbara.
Yes, good morning, everyone. I'm going to share my presentation. Presentation, just a second. Okay. Yes, good morning. My name is Barbara. I'm from ADIS, Portugal. I'm here to present you a little bit of the I34C project on ADIS business case on monitoring, and thank you for this opportunity. I'm going to start by giving you a little overview of the project, talk a little bit about the partnership, then go and focus on the business case, on monitoring, and the instruments that we are developing within the company that will be specifically applied in this business case, and then I will finish with some considerations. About the I34C, this is a project that aims at boosting the macroalgae sector. It aims at providing a transformative and transnational value chain of products for biotechnological applications. It has a component directed to the seaweed-based applications that can go from the cosmetic sector, biofertilizers, food sector, where we have all these partners that we will be working on it, and on the seaweed production that will be very targeted to the pigment extraction, and there is where our business case will fit in, mostly applied to the monitoring on the algae production for all these purposes. This is the consortium that is involved in a project where we are also partners with Submariner. It's from all over Europe, and here just to talk to you a little bit about IDIS. This is our first meeting, so just to present this a little bit. We are an engineering company. We act in these four main markets, so ocean and offshore renewables, blue growth, ports and river logistics, climate resilience, and sustainable water systems. And we try to have an approach that is very related to nature-based solutions, and trying to provide projects that are sustainable and regenerative. Going back to the projects, the I34C has 6 main pillars. The digital solution for open sea aquaculture that we will be developing, it's centered in pillar 2, which consists in the implementation of a portfolio of investments across the whole seaweed value chain. And what is it exactly? The case study, the monitoring case study that we are implied in. So the idea is to create a tool, a digital tool that will provide, that is based on advanced technology to be applied in traditional aquaculture to help with scalability and efficiency, and reducing the uncertainties related to the production of seaweed. The tool will be customized towards responding to the needs of the aquacultures. Actually, we will be we will be going forward with a tool that we are already developing within IDIS, Murex, which is a dashboard that we are already implementing in the scope of various projects, that allows the clients to get access to the data that we produce, or that we measure in real time or in post-processing. So once again, the idea is to get real-time access to the health status of the farm, and to facilitate the interventions that the aquaculturists might need to take, and to help them with planning the operations, so that they can cut maintenance and operating costs, and reduce the unpredictability of unexpected outcomes related to the variability in environmental conditions, and also reduce the operational risks. So this is just a visualization of what the tool is exactly expected to be. So mainly here on the top right, we have the collection of the data that can be either by deployment of instruments in situ or also by using Earth observation data, where we would gather parameters like water temperature, dissolved oxygen, pH, salinity, water wave parameters, and so on. All these data would be processed in this kind of data processing cylinder here in representation, where we would apply machine learning techniques for the modeling. Then there we have these thresholds according to the seaweed tolerances in this case, and that would be from which warnings might be issued if needed. Then all the information is sent to the user through this dashboard through cloud computing. So for this, of course, we have a very important initial phase that is the discussion with the aquaculturists. So really understanding their needs, what are the parameters that they want to have access to, the kind of warnings, the kind of visualization that is helpful for them, for us to really be able to build this customized tool that will actually respond to the field needs, so that they can really help with this operationability of the seaweed aquaculture. Then we will go for the instrument deployments, and further within the project, we hope to go for a stage of scaling up implementation, and then for a pilot and demonstration. And the tool, what kind of services that the tool can provide. So the idea is to have the forecast, so really see the behavior of the parameters that are being measured, try to have enough time of forecast that allows for attention by the aquaculturists to understand if they need to take measures or not, and it will provide anomaly detection, so to identify if data out of normal is being record, and if that data is really a warning to so that the actions can need to be taken, so by default classification. And also it will provide remaining useful life estimation, so that the aquaculturists can understand if equipment needs some kind of maintenance, so it needs to be changed. So these are the kind of estimations that a tool will provide help to the aquaculture farmers. So in the end, the idea is to have an analysis of the predictions to identify potential needs for preventive or corrective maintenance initiatives. And why would this be helpful? Well, we have different levels for action, let's say, and we want to try to go from the reactive response, so the lowest level where we react when something has happened and needs intervention, to go to the predictive stage, so that we can anticipate the needs for these actions, and then increase the efficiency in the aquaculture farm activities. So associated to this, we are also within IDES developing our own instruments. There are low-cost and user-friendly real-time monitoring IoT sensors. So they're supposed to be easy to deploy, with dedicated dashboards, again here connected to the platform that we have been developing, Mirex, and that will provide a global coverage. This is an illustration of what looks like. So we have these buoys that have the instrumentation, and along the cable that is moored, you can have stackable modules with different sensors for the different parameters that might be needed. So here we have the examples that for water quality measurements that can be provided. We can have temperature profiles, wave measurements, and so on. So this would all be incorporated within the monitoring tool for this project. And just to finalize some final considerations, so the I3 Seaweed project, its main aim is to boost the seaweed sector, not only from the production side, where we are involved within our business case, but also to on the ways of transforming seaweed and using it in the different markets. Our monitoring system is expected to be an innovative and advanced tool that will help with the increase of production and facilitate operation and maintenance activities for the aquaculture farmers. So then reducing the uncertainties, trying to reduce the uncertainties linked with the production of the seaweed biomass. And this is very important and can only work if it's created in close collaboration with the farmers, so that we really address their real needs and give a positive contribution to this industry. Thank you very much. That's on my side, and here for your question. Thank you. Sharing.
Thank you so much. We have time for one short question if anyone has anything. Just raise your hand and unmute. Yes, Eva.
Hi, thanks a lot for this really great presentation. I'm kind of algae producer, and I'm very interested in this solution. But do you have any idea if it's already tested for offshore application, or what are the parameters you can use it? Like what's the maximum wave height or water depth or something?
Yes, thank you. Well, actually the monitoring system has been initiated. So its development has been initiated in inland aquaculture, but exactly within I3 Seaweed project, yes, it will be. The idea is to make a deployment in open sea farms. So we are in discussion with other partners from the project. So actually, we will have two testing cases for the implementation. So one is an hatchery. So the hatchery is really it's not out in the sea. So but the thing is that the monitoring system doesn't really care where the instruments are. So it will be flexible enough. So the instruments are there. It will get the data and process it and send it to the dashboard. So we have a natural case, and we will have an open sea farm. That's where we can install the buoys that I have also presented here from Nexus. And technically, there will not be many constraints regarding water depth or wave. It will be designed to the exposed conditions that are expected for an aquaculture farm.
Yeah, thanks a lot. We should keep
Thank
in
you.
touch.
Yes,
Maybe we can test it in our side as
yes.
well.
So may I ask you, where are you based in? Where is your
in the Baltic Sea and in the North Sea as well? You have two sites.
Okay. We are, so our envisaged pilot test, it's Ireland. But yes, let's discuss later. Thank you very much.
Thank you. We're going to move on and open up for Eero, who was talking about blue mission bonus.
Yes.
You want to share your screen? Thank you.
Are you seeing the slide all right?
We see everything. It looks perfect.
Great, thanks. So, hello everyone. My name is Eero Asmala. I'm from Geological Survey of Finland, and I will be talking about the development process of our monitoring framework within the project. First, briefly about our project here. Blue Mission Banos is a coordination and support action project in the EU Ocean Mission, coordinated by Submariner, and we are now on the third year of the project, and we are focusing on the Baltic and North Sea region. And as a CSA project, our main goal is to support stakeholders to achieve these mission goals, and that happens through facilitation of different kinds of activities towards the sustainable blue economy. And in practice, what we do is that we are now in the process of developing this monitoring framework, which then can be used to assess whether the carbon neutrality and circularity are achieved in the future within the sustainable economy. And we are also hosting these mission arenas, which are for us a key forum for connecting with stakeholders and bringing them together, and also showcasing all kinds of solutions for the blue economy goals. And here's below is the link that you can access our project website. And when we started off with the developing the monitoring framework in the project, we didn't just come up with from scratch, but we are heavily building on or leveraging on the baseline study carried out by Technopolis Group. But we identified that as a goal, our monitoring needs should support decision making in a way that it's evidence-based, and it's of course targeted towards the sustainable blue economy. And it should also be something that can be used to track progress, so we're actually going to right direction. And early on, we identified that we should be using the sustainability framework so that we are actually covering different aspects of these in the monitoring activities, namely environmental, social, economic, and governance aspects or dimensions here. And we chose to use key performance indicators as the tool in this monitoring framework, and they were selected because they provide this kind of clear and measurable framework that can be used. But also KPIs are flexible in that way that they can be used across different sectors and the sustainability dimension. So they allow this flexible way of us to do monitoring. And in the Blue Mission Banos project, we narrowed down to 5 sectors, which we identify that these are some of the key sectors in the blue economy that actually hold the key for making it. So in that sense, monitoring the activities and progress in those sectors is essential, and therefore we focus on those 5 sectors there. And in addition to the sector focus, we also all the time carried the sustainability framework with us, so that we would look at these sectors from different societal dimensions, so that we would have as wide scope as possible, but still keeping it realistic and something that we actually can do. So how we actually did it is that we first started off with this desktop study that we collected 419 existing KPIs that could be potentially used in the Blue Mission Banos project to track our progress. And from those 419 KPIs, then through stakeholder workshops where we had experts from different sectors present, 1 in-person workshop and 2 online meetings, we then validated and refined those KPIs so that we came up with a short list of KPIs. And for that shortlisting, we had multiple criteria. For instance, relevance, so that they should be relevant to the sustainable blue economy goals, so that they are actionable in a sense that they they are actually something that can be used to change things, change how we do things, and data availability, that is a key point here, that we try to select KPIs that we know that there is some data at least potentially available that can be used to trace the progress. And we ended up with 50 KPIs, 10 for each sector. And these KPIs, of these 50 KPIs, 6 were cross-sectoral, meaning that they could be applied across different sectors. So they were so broad in their description that they could actually be used in many of these sectors. But most of them were actually pretty sector specific and could be applied only for that sector that in question. And from the 50 KPIs that ended up in our final list, only 18 could be identified with a data source, and data source being something that's updated or something that's collected already, or something that can be used to trace progress. And that varies a lot across sectors, meaning that some of these sectors are have further progress in collecting the data or giving out the data, whereas some sectors still have challenges with it. And our experts evaluated their readiness level in a sense that how ready these KPIs are in using them as monitoring tools. And many of them, like grand average was something around 3. So, but we actually could identify some KPIs that can be used right away to monitor the progress here. So I'm kind of narrowing down to data availability challenge, because that's a very acute question for us now when we are in that stage of the project that we have established these KPIs, we have developed the framework for those, but we are now trying to actually establish the baselines. We are trying to start tracing the progress we're doing. And it's now an apparent challenge that for many KPIs, we're still lacking the data, and we are thinking about different ways to how to actually implement the KPIs, even though the data access is a challenge here. So there are technical issues with the data availability. So some of the data is collected, but it's scattered around in different spreadsheet or PDF files or in different project or something, that it's not collected or merged into a data set that covers the whole Banos region. In best cases, there is a web plugin that we can actually tap right into the data source and use it almost in real time. That's of course the ideal case. But on the other hand, for some KPIs, we are even lacking the baseline, so we don't know where we are now or where we were in the past. So we don't have a starting point. So that's definitely an issue when we want to think about how to make progress if we don't know where we start from. And this calls for regional harmonization. So it means that like different countries, different projects, different regional actors would benefit from harmonizing their ways of what type of data they would be collecting and how to actually store that data and make it accessible. And this means and requires essentially collaboration both across sectors and within a sector, but also across countries and regions. And as a action point, what can be done is that we as a community, we enhance our data sharing platform. So we make the data available in different formats, and our Wavelinks web page is an online platform in process. It's been developed right now, and that will in the future be a site where we collect some of the KPIs and some of the monitoring data as a showcase to what can be done with the KPIs in this sense. And then there could be regional agreements, how to harmonize the data, how to actually make sure that same units and same metrics are being used to measure the same thing, for instance. And then, as we have already identified multiple data gaps, different projects, different research activities, different monitoring activities could be then started to actually fill these gaps. And in a practical sense, it would be great to engage with people who are collecting primary data, who are involved in data collecting in the upcoming mission arenas, which are our prime forums for discussion in these matters. And the next one will be in April, May time in Sopot, Poland, and the following one in autumn in Copenhagen. So these will be very nice venues for sharing ideas about monitoring and the data needs there. And also, we are really eager to get feedback about our KPIs and hear suggestions about what could be monitored further. So I'm sure that we are missing some essential here. So what actually could those monitoring needs in the future be? So with these words, I finish. Thank you very much.
Thank you so much. We have time for a question. If anyone has something, just raise your hand and you can unmute yourself. No questions. Then I think we have a little, oh yeah, Talia.
Yes, hello. Thank you very much, Eero, for the wonderful presentation and also a previous one. I have also a question for both of you. As all this monitoring and data information, they are super important at European level. And my question would be, what is your exploitation plan for, thank you, Barbara, for integrating to the EMODNet and Copernicus, especially EMODNet with the models? Do you have, what exploitation activities do you plan to do for integrating your results to EMODNet European services?
Yeah, thanks. Maybe I can go first. So our partner SDU is developing this wavelengths platform, and that will be one of our key technical features. And as I understood, they have some plans of tapping that wavelengths platform also to other data sources than what we are now collecting within the project. But I'm not sure which ones are specified there, if EMODNet, for instance, is one of those. But I think the plan is so that the platforms can be like the data sources can be varied within those platforms, so they're not fixed for one data source.
Yes, maybe I can also say something on that. Thank you, Talia. Well, so the tool that we are developing is based on machine learning modeling. So the more data we have, the better. And yes, it is our intention to have the instruments on site, which is really important. But of course, then the data length of the time series, the data availability will be quite limited. So having other sources of data is also very important. The only issue that we need to be careful to pay attention to is the resolution of the data that will be incorporated. But this is something that indeed we want to integrate in situ measurements with other more globally available data for the sites, so that we can have the most robust possible monitoring system.
Yeah, this is very interesting. And we're expecting also Maya, also the digital twin of the ocean. That will be a revolutionary approach, but we are still working on, they are still working on the specificities, but we expect to have a lot of interesting integrations there that will be the one-stop shop for all this monitoring and data that is produced there with many functionalities. But let's see what's going to happen. Thank you very much.
Thank you all for questions. We are right on time, so we're gonna take a 5-minute break for everybody to get some coffee or a technical break. And then we will be back at 10 to look at some monitoring frameworks and needs. So I give you 5 minutes and then we'll be back. Okay, our 5 minutes are up. We are gonna go on to the next section. So now we have been talking a little bit about kind of monitoring markets and some monitoring business cases that are going on, and we're going to move on to our monitoring frameworks and needs, starting off with the Coolblue project. And Sonny, are you here?
Hello.
Hello. I'm going to stop sharing so you can share your screen.
Yes.
And I hand over to you.
Can you see the full screen or you just see part of it?
No, it looks good.
Looks good. Okay. So my name is Johnny. I come from Greece and I did my master's in University of Gothenburg. And I did my master's with the Coolblue project. And today I'll be presenting about the monitoring methods that we use for monitoring the low trophic aquacultures. So what is the Coolblue? The Coolblue is a collaborative initiative which is aimed at establishing a network of about regenerative ocean farming in the Northern Europe, particularly in the Nordics, but also have the second branch, let's say, which is about the Baltic. And secondly, we are aiming in order to promoting about low trophic aquacultures, such as seaweed and mussels. And we also try to promote about scaling out instead of scaling up, which we aim to have to promote about having to exploit the area so we can have multiple small scale farms instead of a few large monocultures. We can exploit one large area, which also results in higher stress to the environment. And this our thesis was included about the work package 3 in the Coolblue missions, particularly 3.3, where we used about video monitoring and eDNA sampling. And because every time we have some sort of operation in the marine sea, such as having some structures like for the mussel or the seaweed aquacultures, any modifications to the local ecosystems can have also potential habitat modifications because we introduce these new artificial structures. And in order to address this and to see what is the effect of these structures to the environment and if it benefits positively or negative or it's neutral, that's why we want to see using non-invasive methods in order to see what's happening down there when we don't see anything. And for this reason, the monitoring methods that we used was environmental DNA and video monitoring. So today we'll be presenting Mats Heitman research study that he did, which include about the eDNA using water sampling and also using the autonomous reefs monitoring systems. And also we'll be talking about my project, which was about video monitoring, which using a bait. That's why we call it a BRAV in the marine science. And also how it performs when we include the machine learning part, where we are able to automatically detect and classify species. So the study objectives have been to use the eDNA in order to be able to detect the species presence, which species we find around, and about the composition when we compare it with different sites where we sampled. To use the video monitoring, including the machine learning, in order to analyze the species, what species we find, the richness, the relative abundance, how many of them we find, and also how it compares when we compare the sites where we sampled. And the reason we do this because we want to have like a sustainable monitoring strategy and system for regenerative ocean farming. So we can have an insight about what's going down there. So concerning the eDNA sampling, Mats, he sampled in 3 locations. On the left, you can see the map where are all the locations and which method we used. In Helsingborg and Copenhagen, it's the place only Mats sampled with eDNA. And I sampled with video monitoring in the northwest in Lysekil. And Tjörn was the only site we had combined up where we both sampled with BRAV and with eDNA. So Mats in total, he deployed 14 arms, those artificial structures that I showed before. And he used these 3 farms, and for each farm he had like triplicates or duplicates of those structures where, for example, in Helsingborg, he had 3 arms in the farm and 2 arms in the control zone. And below you can see also the picture of how those arms look like before the deployment and after the deployment. In the case of Mats, he didn't deploy it down to the benthic. He kept it suspended in the 2 meters below the farm. So there we would gather all the DNA. So the way it would work, like for example, Mats, he would get the arms after few months of deployment, and then he would scrub the DNA and he would extract it. And in order to match what species are found, what DNA is found in those structures, he used the COI library, which mostly targets the invertebrates, and the 12S, which target the fish. And the process of DNA is like you collect, you filter, you extract the DNA, and then you amplify and you sequence it, and then you compare, you use those pipelines in order to match what species are found. And his results are, for example, in the first graph, we see about what species are, how many species are found and in which location, which F stands for farm and C for control. And we can see that when he used about the COI library, where it's focused about invertebrates, we see that he found that there were more unique invertebrates in control zone, which could explain that because in invertebrates it's like a lot of filter feeders, so they come in. For example, we farmed also only mussel aquaculture, so it's one hypothesis could be also because a lot of filter feeders, they might compete with the filter feeders of our farm. And instead, when he compared about the fish communities that he found, we see that we found more fish in the farm, more unique species of fish in the farm sites, which also could be explained that for example, the mussel aquacultures, they can provide shelter and also food for the fish, and that's why it could be explained why the number is higher over there. And also out of the species that were found, it's important to note that he detected also one critical endangered species, which was the European eel. So we can see the importance of the eDNA, that even though we don't see anything, we can detect a lot of cryptic and rare species. And in the middle figure, we can see combined PCOA plot about the commute composition between the sites and the farm and the control treatment. So we can see that with the green color, we can see the species that the composition of the farm, and inside it we can see also the composition of the control. So we can see that they both overlap, so they share the same species composition, like the species that we find in the control zone, we find also in the farm zone. But also here we can see, if you see the dotted lines, the black and yellowish, it's the Copenhagen and Helsingborg. We see that it's clustered together also because they are very close in distance, and because they are both in the harbor, so it makes sense that they share the same species composition. And in the other cluster, we see that it's Shorn, which also shares a different species composition compared to those sites, and which also makes sense because was a bit further. It was not in the harbor; it was also in a sheltered area in the marine ocean. And we can see how easily just with this basic system, we can get this valuable data and how they cluster together. And also it makes sense because making also from distance and what species they collect. And here is my work that I did with the BRAV video monitoring. As I mentioned, I sampled only in the west coast of Sweden, particularly in Lysekil and Tjörn. In the second picture, you can see the setup that I used. It was more like a DIY setup. I just custom made it. And you can see also what is included there, very basic stuff. And I monitored in each farm, and I dropped the camera once in the farm and once in the aquaculture for every site. And I filmed 30 minutes in each. And on the right, you can see how the camera view is when we get the video, and we process it later on. And my results in the left graph, we can see the min-max n, which corresponds to relative abundance. We can see how it fluctuates per month. And we can see at the end about when we compare the mean values between control and farm, which were very close and not statistically different. And the same thing with species richness. We can see how it fluctuates every month. And when we compare the mean species richness per site for control and farm, we see also that it's quite similar. And when we run again also the PCOA plot about the species composition, we see that the species composition that we find in control zone, we also find in the farm zone, which shows that there are not much difference on what species we find where we have the aquaculture practices and in the sites where we have the control, where it's a bit more distant. And what I did was I want to see also how we can combine my data and Matt's data, eDNA and video monitoring. So I included the two locations where are very closely related, like Lysekil and Shore. They're in the same area. So I did compare about the species that he found about the invertebrates, they're detected both by eDNA and BRAV, and we see how many they were detected by each. For example, we can see that the swimming crab, the gastropods, and shore crab were detected by both eDNA and BRAV. But we see that the shrimp, hermit crab, and spider king crab were only detected by BRAV, but they were not detected by eDNA. So one explanation for this could be, for example, for shrimp, they don't shed a lot of DNA, so probably that's why it was not detected from the ARMS project. But the hermit crab and king crab could also be not detected because, for example, the libraries that they have when they match species, it's like a big database, so they don't have all the species. So maybe that could be one limitation why these species were detected only by BRAV, but not by DNA. And when we compared about the fish communities that were detected by BRAV and eDNA, we can see the overwhelming advancement of eDNA, that it detected 92 of the species, and only 2 taxa were detected by BRAV, which also could be affected the way I designed the system, like because I was filming downwards, so I reduced kind of the field area about how much I can see in the water. But of course, my setup was focused mostly on the invertebrates and the epibenthic species. Maybe one thing that I should note here as well, it was like when I included invertebrates, I removed, for example, copepods. I removed also, because they're very small size, I removed also worms. So in order to compare this, I included the epibenthic invertebrates, which are also in the size that you can see also from video. And because with eDNA, you get a lot of invertebrates, but you get a lot of small size, which you will not be able to see with the video monitoring. So it does make sense to include when you compare these two methods. And here is when we apply the machine learning part. So this model and dataset, it's publicly available at Zenodo. And if you scan the code on the right, you will be able to get redirected over there. And you are able to see all the pictures that were used for the data training and testing. And also, if you are aware, if you are in collaboration with somebody that they use this type of running this type of models, they can just download our model and run it. But of course, it will be more efficient. This model will be more efficient when you have a similar setup. But of course, people can take this data set that we have created and we have trained the model, and they can also include pictures. So we can have more pictures of Hyesaraneus, more pictures of Pagurus, and they can, and eventually you will end up with a big library where you can have the model for every species associated with the farms. And in order to run this model training, I use the Swedish platform for subsea image analysis, otherwise known as Subsim. And what are the key findings about these two monitoring methods? It was that eDNA is very good when it provides species richness, including the detection by for critic and rare taxa. And also, it's the positive is that eDNA can capture herbivore species that the BRAV may miss. Because BRAV, I use a bait in order to track the fish and all the taxa over there. So the herbivore species, they will not be attracted by that. So the BRAV, it's a bit biased towards that only opportunistic and carnivore species will come there. So you miss out on something. But when you use the BRAV and the machine learning part, the AI, we are able to provide abundance about how many species we find and when we find them. And also you are able to get species that they don't shed that much DNA, which are unable to get it from the eDNA part. And when we combine these both methods, you get the hidden diversity, and also you get the, again, presence and abundance trends. Some of the limitations were like the DNA drift with currents, so we cannot pinpoint the species that we find with eDNA. We cannot pinpoint them in the map where exactly they were. Also, the complete reference databases. It's about that they don't have all the species in the database. So if the species not included in the database, you will not be able to match it. And also the BRAV, it's biased towards the carnivore species. And one suggestion, in order to close now, one suggestion would be this: when you run such monitoring methods and fieldwork, it's very important to have the seasonality when you farm, when you field. For example, here we can see the graph about the gobies, about the fish, and how they migrate during the quarters of the years. For example, we can see that in the winter, in the first quarter, it's in deeper waters, and then during the summer, it's in shallow waters. So in my case, the film was in the cold months, Q4 and 1. So we see that it was not optimal in order to obtain the most out of it, like to get a lot of species. And also our fieldwork with Mats were not overlapping, because I filmed during the winter and he filmed during the summer, so there's a bit of gap over there. Second important, it's the consistency. If we want to have a monitoring, if we want to get results and to have concrete results, we have to have long-term monitoring, not just one time. And that so we can have for longer periods, and we can see how it fluctuates through the year. And ideally, we would like a before-after control impact, because now that a lot of farms are created and they build up, And it's a really good chance that we can have these monitoring methods and we can have them before they are, since they get the permit, we can fill them before they're installed, during the installation, and after it runs, so we can have a full picture of what's going on before the installation and what's the impact on the benthic species after the installation of those farms. And also we can utilize the AI for also size estimation, like those boxes that you see, that we can estimate the size of how big is the fish. So this way we can get also the biomass, but also compare, like, do we get bigger size fish in the farm or outside the farm? And also we can use the AI part in order to assess the behavior, how it behaves when it comes in, when the species come over there. And thank you very much, and looking forward for any questions.
Thank you so much, Sonny. You used your time and a little bit more, so I think we're going to save the questions for you till the discussion. If you have anything, you can also add it in the chat.
Yes.
And now we're going to go on and we're going to listen to Sophie Koch from Sjökovin and Seamark.
Yes, thank you. Can you hear me well?
Sounds good.
Good. Okay, I'm sharing my screen. There we are. Can you see it full screen?
Yes, it looks good.
Perfect. Thanks. Yeah. So super interesting to listen to the calls that to the talks that we had so far. And my talk is actually quite connected to some of them. For example, this morning we heard about the key performance indicators. So I'm going to also talk about some indicators that is linked to that. And when Barbara, I think, was talking about how monitoring is crucial to the increase of production, this is exactly where my talk adds on. Basically, it's an assessment on what we have to watch out for when we're expanding. And I'm going to talk to you about what carrying capacity means and the expansion barriers, basically, and what we need to monitor in there. So this is part of the Seamark project. I was able to integrate my PhD research in there. And this was also to bring seaweed products to the market, but also to look out what happens when we upscale. So quickly, what I'm going to go through is basically, which is the main key of my presentation, the difference between what you monitor, what's going on right now in your cultivation. My talk is all seaweed based, by the way. And then also what you need to monitor if you are talking about expanding, so upscaling, intensifying, and so on. And theoretical framework that we created to assess that and what is to monitor within that framework. So monitoring the status quo, you're all familiar with it. It's sort of, checking, are you not doing any harm? Are there any negative impacts from your cultivation, any type of cultivation, but in my case, seaweed, you do it either like once a year or continuous, and sometimes it's state demanded, like you have to prove that you're not doing any harm to the other species of the ecosystem and so on. But you could also do something like monitoring of ecosystem services, which is what we've heard about just now in the talk. Studying biodiversity, how is it behaving? Are we having a positive impact? And this is not something that you have to do, but it's super beneficial. Once you know this, you can use it in your business case. You can maybe get additional funding. You can get credits and so on. But for this, you really need to prove what you're doing, and only through monitoring that's possible. So what do you need to monitor when you expand? So why at all looking into this? Because to put it really simply, if you're intensifying, if you're expanding, if you're upscaling your aquaculture, you're also upscaling and intensifying your potential impacts, positive or negative. So on the positive side, we had all of these discussions of like, how much do we have to upscale the seaweed cultivation so that we can sequester enough carbon and so on. Calculations about this were going on, but sort of not really taken into consideration. There might also be negative impacts. And if we just upscale like this, can we do it without any limit to it? So taking my background picture just as an example, this is in the Faroe Islands, and it's somewhat of a small scale farm. You can see the boat, you can see the few buoys, the yellow ones for that one cultivation unit. Versus, for example, how cultivation can look like in China. This is a picture taken from the Chinese coast. So imagine the difference of impacts you have it. And the small seaweed farm in the Faroe Islands, you could still buffer some of the negative impacts. Also, you have less positive impacts. But still, what impacts are we having with a super large scale farm like we see in the right-hand lower picture? So again, coming back to what can be monitored. So the positive impacts, biodiversity, the bioremediation, the carbon sequestration, and so on. On the negative side, seaweed cultivation has also negative sides. Like there's also the disease that could be coming. Maybe some invasive species could be coming along. Maybe some other impacts, just like shading isn't a problem on a small scale, but maybe on a big scale, if you shade too much, you could have a negative impact on in case you have seagrass below it, for example. So on the positive impacts, I was saying, okay, great. If this is expanding, it's not a problem, really. It's sort of the more the merrier. Obviously, this is really just very simplified. But on the negative impacts, just to make the point, is the comparison, there will eventually be a limit of unacceptable change. Eventually, your impacts may have, I don't know, but so much harm that it's not acceptable anymore. It will cause damage that you consider, that you notice, that you say, we don't want to go this far. And it's the carrying capacity that looks into these aspects. So the carrying capacity has 4 dimensions. Often what's known in like the finfish aquacultures and so on is more like the production carrying capacity. The physical is sort of the first step. To me, it's really in simple words, the body of water that you have is physically limited. There's a bay, there's a fjord, and it doesn't go beyond that. The production carrying capacity really looks at the stocking density of how much fish can you squeeze into this little tank here, or like the big circular nets that you have out in the aquacultures, without having a decline in their production. Like at some point you'll get disease because they're squished too densely, or at some point, I don't know, there's a ratio of food that you're losing and so on. So there is an optimal stocking density. Ecological and socioeconomic carrying capacity are sort of more newer, and the socioeconomic one's very new. Both are looking into the negative impacts that you potentially have, and when are they becoming unacceptable. And what you assess here is really, where are the limits to your expansion? How much can I expand or upscale or intensify until I reach these limits? So, as I said before, for seaweed, there were loads of, in that hype, there was loads of discussions of, we can extend and we can like cover 2% of the ocean. Just, these are not the correct numbers, but there are studies out there like this, but they're not considering, as I said, the limit of unacceptable change that you might have. It's just not been considered. So we looked into this, and we found out actually, before even you get to that point, you have a lot more socioeconomic barriers. And everyone who's familiar with seaweed knows that there are barriers to expanding the aquaculture here in the European seas, or even North America, that hinder the industry from really growing and to getting to its potential. So it could be the market, it could be the licenses, profitability. We're not there yet. It's hindering for it to upscale so that we can actually use all of these benefits that seaweed aquaculture would also have. So I called them the barriers to expansions that actually are way earlier. So ideally, if we were to assess these barriers and then say, okay, we do something about them, and I know that the European Commission is doing something about them already, there's communications about this out there, all of the actors here in the call as well are contributing to that as well, so that we can reach a level or an optimal window, I called it, for like an extent of cultivation that is finally beneficial. It's big enough so we can benefit from the seaweed industry, but still below the limits of unacceptable change. So still below where we create damage to the ecosystem. So how we studied this is a bit theoretical. I'm just going to go over this real briefly. So what we said, okay, there is something coming into the farm. It's like the conditions, the surrounding that you have. It's from the environment and the society. It's the conditions that are existent here in which you place your aquaculture. Then you have outputs coming from the aquaculture. It's like the impacts coming from it to the environment and to the society. And we called the positive and negative input conditions. We call them either the favorable conditions or the limiting conditions, or variables in that case. And on the other side, on the right-hand side, is the positive or negative impacts that are coming from your farm to environment and society. So we studied them, we listed them, obviously on the socioeconomic and ecological dimension. We came up with big lists, but then when we looked at these big lists of positive and negative impacts and so on, we said, okay, the positive ones and the favorable conditions, great, good to have them, good to know them. We can use them later, but they're not the critical points here, because it's sort of the more the merrier. However, if you look at the negative ones, there you may reach limits, or you have barriers that actually hinder you from upscaling. So these are the ones that are really interesting to look at and to study. So created this 12-step framework that's like first have to plan, integrate your stakeholders, and so on. But then in the lower parts, in the blue parts, is where you can look at all of the different those big lists basically that we had, the limiting inputs and the negative outputs, and you say which ones are relevant for my context. Obviously, not all are relevant for every context. And then you say you look at the indicators that we also created and the thresholds and barriers, and you can go about to assessing this to see what do I have to watch out for if I wanted to upscale. So the results looked something like this: 2 big lists where we reduced already the numbers of inputs and outputs, so the conditions and the impacts, to the most relevant ones. And so we have a list of limiting inputs and their indicators, how we would go about measuring them, and the barriers. And the barrier here, the word is a bit tricky because basically it's a barrier that hinders you from upscaling, as I said many times. But you kind of are looking for when is the barrier gone, because then you can upscale freely. And the negative outputs have also indicators and thresholds, and that threshold means don't go beyond me. Like from there, it's been assessed, or it can in every context be assessed and evaluated. This will be unacceptable. And so in pink, we had these socioeconomic inputs and outputs, and in green, the ecological ones. Obviously, it's just a screenshot. The list is much longer. So for my theoretical framework, and since this is a talk about monitoring, I kind of wanted to just clarify that real quick, because I keep on talking about assessment. So the assessment for me is the process of defining what is unacceptable, of defining and identifying where would be a barrier and what is that barrier. What you evaluate there is really like, what's the value of that limit? And for every variable that we looked at, obviously is different. And what's to be monitored here is basically, what do I have to watch out for? What are the indicators that I have to keep on measuring and monitoring to know I've reached the limit, or to know I'm finally past this barrier? And just a couple of examples. Some of the limiting conditions, let's take my study, like the experts that I used in the study. They were saying, for example, okay, there are too high operating costs. This is really limiting us to actually upscale successfully. And the indicator that they said how we measure this is, yeah, we watch out for the cost benefit of our company, and we still see operating costs are still too high. The barrier that you want to overcome is once you finally reached the profitability. Very similar with the third one, there is also socioeconomic ones, is the lack of investment. And they say, yeah, without the investment, we can't upscale the way we want to. Indicator monitoring, the indicator to measure would be, for example, is there governmental support coming? And one of the suggestions for the barrier was, once CVID is incentivized, well, that's great. Then the lack of investment isn't such a barrier for us anymore. Or on the ecological side, let's say you have lots of fouling and grazers that actually reduce your production that you have at the end. You know, once you harvest, you might have such fouling going on that you reduce, that you miss some of the biomass. And the way you monitor this, or the way you measure this, would be then, yeah, the growth rates, the effect that it has on the growth rates. And then the barrier that you want to overcome is, great, we don't have fouling anymore. A bit more accessible, actually, a bit more easier to understand are the negative impacts. So, as I said, seaweed farming has a lot of positive impacts, this ecosystem services, but some of the negative ones could be every activity has pollution, probably. There is, you know, plastic that's being shredded off the cultivation lines and so on. And what you want to measure is the plastic level, and the threshold. They was that actually the threshold is at 0 or as low as possible. We don't want to shed anything into the environment. So it's very, very low threshold. Or for example, something like shading, the sunlight that is captured by the cultivation and then doesn't reach the bottom anymore. So there's a competition for the sunlight. And what you want to measure is really the amount of shading that you have. And then just suggestions for thresholds would be, you know, compared to a reference site, we don't want to be shading more than 30%. So we actually, you know, give other species still a chance, you know, phytoplankton swimming below and so on. A bit of the socioeconomic negative impacts. imagine you have a seaweed farm and you haven't talked to anybody around, and then there's opposition from fisheries saying, wait, we can't fish anymore here because you're now with your lines in our water, and they're against it. And then so what you have to watch out for, the indicator that I said, is check the social acceptance. Check if everyone is still accepting that you are here as a new player in the coast. And the thresholds are very context specific. And what we just said here is community set the control points, because obviously it really is like a discussion going on. Is the acceptance right there, or is it, nope, this is unacceptable right now, we want you out of the coast? And then obviously something has to be done. It very similar to this is the user space needed. The coasts are very full, and then seaweed company maybe comes if it got a license, and then other people are complaining they didn't get the license, and there's lots of needs in the coast basically, and you have to sort of measure these different needs and the complaints, and then the threshold that I said, once you've reached a threshold, once you've expanded too much, is when really there are incompatible user needs. So it's still a bit complex to grasp this, but what I want to get to is basically that when expanding, it's sort of all of these indicators that have to be monitored to see if you're getting to a point where it gets unacceptable. And obviously the limiting conditions is way easier, because if you ask any seaweed company, this is exactly what they're watching out for anyways. They will be monitoring, am I cost efficient already? Am I having any difficulties with, am I losing any of my biomass due to, fouling? This is exactly what I watch out for, because they want to grow naturally. And but it's more not so much on the radar of people yet, is all of these negative impacts that you have if you want to upscale. You have to monitor yourself. Are you reaching any of the unacceptable levels? And yeah, this is my last slide, basically just summarizing why we need to monitor this. So the very first point is exactly what I said before. People or companies will be monitoring already for what they know are their barriers. But it's just more interestingly to understand where the barriers are. And then also just this new aspect of this carrying capacity stuff is that it is interesting and important to watch out and to know where the thresholds should be and keep on monitoring so you don't overstep them. And this, I think, goes really well or integrates into the other frameworks that we've heard before from HIDIS, from Barbara, or the key performance indicators is very linked to this. It's just a tiny different angle to it. And I think that's the end of my presentation. Thank you.
Great, thank you so much, Sophie. We're going to try to stick to the agenda and take all of the questions in the discussion. But I think we'll have some interesting discussions also about eDNA and fouling with our last presentation. But now we're going to go to Michaela from Aqua Monitorix.
Hello, thank you so much. I'll just share my screen.
Great, it looks good.
Perfect. And you can see my title slide there, yeah, full screen?
Yeah, it looks good.
Perfect, lovely, thank you so much. So, hi everyone, my name is Michaela Hogan. I work within the R&D team at a company called TELLab and its spin-out company Aqua Monitorix. So I'll tell you a bit more about that in just a moment, but basically I'm here today to talk about what are the benefits of continuous monitoring of nitrite and nitrate in RAS systems specifically, and what our analyzers can potentially provide for your facility. Now, I'll preface this by saying I'm no expert in aquaculture. My role is more on the technology development side of things. So I'm sure I won't be telling any of the aquaculture experts here anything they don't already know. I've really enjoyed the presentation so far, but some of the technical stuff is kind of going over my head a little bit. So I do look forward to the rest of the presentations, though. I feel like I'm learning a lot. But I have quite a few slides, so I might talk a little bit quickly. I'll try and get through them because I know we're short on time. So yeah, I'll go ahead and get started. So, as I said, I work for TELLab and Aqua Monitorix, which if you haven't heard of either of them before, you have now. TELLab is a privately owned Irish company, and we've been in business for over 30 years. We have lots of different departments and laboratories here, but mainly we focus on environmental analysis, oil and fuel analysis, and chemical production as well. So as I said, I work in our R&D department, which is once upon a time where Aquamonitrix was actually born. So Aquamonitrix was established as a separate company, spun out from Tel Lab in 2021. We are a technology provider in the water quality space, and we develop analytical instrumentation for real-time in situ monitoring of water across a range of applications, from wastewater to aquaculture and drinking water applications as well. So to give you a brief overview of what I'll be speaking about today, first I'll talk to you a bit about our technology and explain how everything works and how the Aquamonitrix analyzers are different from current technologies being used in RAS facilities at the moment. I'll also discuss the benefits of continuous nitrite and nitrate monitoring and why it's so important to measure these parameters in RAS specifically, as well as the value that these measurements can add to the sector. And finally, I'll show you some case studies and examples from data from some of our clients to illustrate how they're using the analyzers to optimize their facilities' performance and protect the health and well-being of their livestock. So when I say current technology, I'm mainly referring to the use of UV probes and other devices employing UV spectroscopy. So there are two main issues to touch on here. Number one being that the UV probes aren't selective for nitrite and nitrate, and their performance is quite negatively impacted by fouling. So they require a lot of maintenance and recalibration, and this obviously incurs additional costs in terms of time and labor. Number two then is that UV methods of detection are impacted by varying salinity levels, which can obviously be quite a problem in an industry like aquaculture. So these devices typically work via different algorithm adjustments and rely on matrix-specific calibrations, which can have a negative impact on the accuracy and reliability of measurements. In terms of colorimetric methods of detection then, there are handheld analyzers based on colorimetry, but they're manual and they deliver quite poor accuracy. So this ties into the third issue listed on the slide here, which is that colorimetry like UV probes can also be impacted negatively by salinity, but also turbidity and organic interferences as well. So our method is based on ion chromatography, and it has numerous advantages over these types of methods. So there's no requirement for multiple reagents, there's no sample pre-treatment, we have high selectivity even in complex samples. And it's worth mentioning here too, something that we probably all already know, but another option is manual sampling and lab analysis. But this grab and lab, it's not really sufficient to capture trends and significant events. They're often missed in measuring in this way. So we won't even mention the cost and the time that this requires. So just to keep that in mind as well. But this slide here just highlights some of the points I've mentioned already. So you can see here on the left how the lack of selectivity on most UV devices makes accurate determination of nitrite and nitrate quite difficult. On the right, you can see the same kind of a curve, but now you have the added impact of the 30 ppt salinity, which sends the measurement through the roof and obviously wouldn't be an accurate representation of real nitrite or nitrate concentrations. So how are these issues overcome by the Aquamonitrix analyzer? Well, firstly, I'll give you a quick introduction to the system in case you haven't seen or heard of it before. So here you can see two different versions of the nitrite and nitrate analyzer. The one on the bottom here looks a bit like a suitcase, which is quite fitting because this is the portable version. So above that is the wall-mounted fixed version. So both employ the same technology, it's just depending on what you intend to use it for, which one might better suit your needs. So the wall-mounted version tends to be more popular for, say, process control applications, and the portable one, for example, is quite useful if you have multiple tanks or species that you want to sample from. So speaking from my own experience, I deployed one of each of these analyzers to an aquaponic facility in Spain last year, where the owner liked to move the portable analyzer between his tilapia fish and his shrimp tank. So that was from a freshwater to a fully saline matrix. And this wasn't a problem for the analyzer because it's not impacted by salinity in the same way that the UV probes are, which I will explain in more detail in just a moment. But yes, so that made it very easy for him because all he had to do was take the analyzer's sample inlet tube out of the fish tank and place it into the shrimp tank. So there was no fuss about it from his end. Varying salinity levels had no impact on the analyzer's precision or accuracy. And yeah, so the analyzer, just to go through the points here, the analyzer employs rapid ion chromatography with patented UV LED detection to achieve simultaneous and selective detection of both nitrite and nitrate in situ. It's proven to be more than 95% accurate, and it is, of course, ISO accredited as well. So how does it actually work then? Well, as I mentioned, it's a method based on ion chromatography. And if we check out the fluidics diagram shown here, we can see that after automated injection of our sample into the system, it's carried along by our mobile phase, or our eluent as it's labeled here. So it's carried to our anion exchange column, where we then get separation of nitrite and nitrate, followed by simultaneous and selective detection of both analytes on the patented UV LED detector at 235 nanometers wavelength. So what we end up with is an accurate and reliable and robust measurement for both nitrite and nitrate as individual concentrations. So you might remember this figure on the left from a few slides ago, but to compare how our analyzer measures up versus the UV probes I was talking about previously, we can take another look at the same absorbance profile and compare it now to a chromatogram from the Aquamonitrix system on the right there. And we can see the difference the separation makes to the ease and accuracy and reliability of measurements. So by employing ion chromatography in this way, we can achieve separation of nitrite and nitrate, and then we're able to measure both species directly. So I won't bore you too much with the details, but coming back to the point I made earlier about why we don't experience the same issues with salinity, it's thanks to the matrix elimination effect of using sodium chloride as the mobile phase for our IC system. So as you can see here, we have a comparison of measurements from samples ranging from fresh water on the bottom up to 20 ppt brackish water, and finally to fully saline 35 ppt water. So absolutely no negative impact on detection of the analyte peaks, as you can see, no loss of accuracy or precision. And also I'll mention here that the analyzers are calibrated before they leave our factory. So that calibration holds true in any sample matrix from freshwater to fully saline. So there's no need for any kind of recalibration or adjustments or any additional time or effort out in the field. There's no extra effort required to ensure that the measurements remain accurate and precise. So this is important to note because in the past, I believe we had actually clients with freshwater facilities who were required to dose seawater for different reasons. So while that'd be an issue for other UV devices, with the Aquamonitrix analyzer, you can sample in any level of varying salinity with no problem. So what does all of this mean for RAS then? Well, with a knowledge of real-time accurate nitrite and nitrate levels within the system, we can achieve an improved understanding of the biofilter status and at startup phase throughout production. We can also improve our estimation of maximum feeding rates towards biomass gain and feed conversion efficiency, as well as better identify when we're feeding in excess. And finally, we can gain a better understanding of our water quality. So this would not only fulfill our moral obligation of ensuring the well-being of stock and avoiding nitrate stress, but also reduce the risk of mass mortalities from nitrite toxicity. So in other words, you big, happy, healthy fish, so they're bigger and tastier then at the end of the process as well. So next I have just a couple of slides and results that I'll talk through briefly, a couple of these case studies. So we have some freshwater and fully saline RAS facilities in Ireland, Norway, Canada, and the Netherlands. So first up is Ireland. Here we see the data from an Aquamonitrix unit that was deployed over 6 weeks in a freshwater RAS facility. So the data was collected post-biofilter to monitor changes in nitrite and nitrate concentrations, while the RAS facility at the time was stocked with about 9,000 Atlantic salmon. So you can see on these graphs the red triangles indicating grab sample results, which are quite comparable to the analyzer's in situ results. But while this provides evidence that the analyzer was reporting the correct concentrations, the drawbacks of grab samples from the monitoring of these analytes in RAS, it can't really be ignored. And I've touched on that already, so I won't go into it again, but you can see a good example of that here on the graph on the right, where you can see a spike of nitrite during week 2 of the deployment. And you could see how an event like this could have easily been missed if you were relying on grab sampling alone, or even alternatively, that peak could have been detected at its highest point, which would also be an inaccurate indication of the overall concentrations. So all of this is basically just to say that the continuous monitoring of these parameters really is what's required to generate useful data and a better understanding of the bigger picture within a RAS facility over time. So here in Norway then, we had 2 RAS systems running in parallel on the same site, started at the same time. However, in one of the systems, there was an issue with the biomedia and the membrane of their biofilter. So as indicated by the rising nitrite levels, you can see it highlighted in blue there on the top figure, the darker blue color. So to fix this, the client took a sample from the biofilter of the working RAS system and dosed it into the system with the issue. And after they did that, you can see how the nitrite levels started to steadily decrease back to a normal level again. So this is an example of how, through the use of the Aquamonitrix analyzers, we can facilitate quicker corrective action over issues like this that can arise. We also know that one of the most key phases in RAS is the filter startup. And the data here shows just how important it is to monitor continuously during this phase, as we see that there can be different nitrite and nitrate levels even for theoretically identical systems. So in Canada then, you can see the levels of nitrite that we started out with, which were elevated and then corrected simply by dosing with fresh water. So again, just to point out the value of real-time continuous data, capturing these kind of trends, and otherwise you'd just miss these kinds of pollution events or you'd catch them too late. And then this is an interesting one. So not to go on about salinity again, because you're probably bored of hearing me talk about it already, but this is data from a fully saline RAS facility in the Netherlands, which was monitored. In this facility, we were monitoring yellowtail kingfish, and that was pre-biofilter. And again, with some grab sample results indicated there in the red triangles. But actually from this same client, we also have some data on their final effluent monitoring. And this client actually had already met their 2025 sustainability goal of a 25% reduction in residual waste per tonne of growth, which is in line with the United Nations Sustainable Development Goals. But to highlight here that the monitoring of these parameters in final effluent is particularly important, especially for RAS facilities in countries like the Netherlands and Scandinavia, where nitrite and nitrate discharge limits are quite strict. So for example, in the Netherlands here, with the country being so flat and they tend to have issues with eutrophication, to manage this, there's very strict effluent legislation that must be adhered to. So the main takeaway for clients like this one is that they're achieving significant savings through the use of our analyzer because they're no longer spending such huge sums of money on grab sampling. And they're also ensuring that they're in compliance at all times. So it's a win-win situation, really. And then just to mention that all of this information is fed to our online platform called Data Monitrix, which you can see it here in an example of the dashboard. The data is communicated via SCADA and IoT, and you can have an overview of your nitrite and nitrate measurements in milligrams per litre as nitrite and nitrate, or as N. So it's whichever you prefer. But the platform makes it easy to spot these trends and catch pollution events at a glance because you have this remote access to your real-time concentration data. And you can also set up alerts, alarms, thresholds, so you'll be notified and you can take immediate action if the nutrient levels do become too high. So to finish then, our main takeaways are that our water quality has an impact on our fish quality. Due to the toxicity associated with nitrite and excessively high levels of nitrate, fish health and growth can be negatively impacted. So with a greater understanding of our water quality, we can reduce the risk of mass mortalities and fulfill our moral obligation of keeping our fish healthy and happy. We can also gain an improved understanding of biofilter performance at startup phase and throughout production by knowing our nitrite and nitrate levels accurately and in real time. So this can assist in making operational decisions about feed towards optimization of biomass growth. We also have the potential for improved estimation of max feeding rates and to identify cases of excess feeding. So this obviously incurs significant operating costs, and any leftover feed not eaten by the fish will increase nitrogen waste within the system. So in conclusion, just accurate monitoring of nutrient concentrations in RAS systems during the whole production cycle really is essential to maximize profitable production, and this can be achieved using the Aquamonitrix analyzers. So that's all from me. I hope I didn't go too far over time. Thank you so much for listening. And if there's any questions, I think we're going to wait until the Q&A. Is that right?
I think that's the best way to do it.
Perfect.
So that we'll try to keep the time. And we have our last presentation before our Q&A and discussion. We have Kelm Young from Glasgow University, who's going to continue talking a bit about eDNA.
Hi. Are you hearing me okay?
Sounds good.
Yep, I'll just share this. Let's hope this works. Are you getting the wrong one?
We see your presentation mode thingy.
Bear with me. Sorry, Maya.
No problem. Now we just see this.
Okay. Let me try. Oh, good. Still.
Yeah, no worries.
Do you want to do it from yours, Maya?
Yeah, sure. I will pull it up, and then you just let me know when you want me to change.
Sorry, thank you.
1 second. No, I just have to find it as well. Let's do this. And we have it here. And this should be the full one, right?
Yes.
Perfect.
Okay. Thank you, Maya. Sorry about that. My name is Cam Young. I'm a researcher at the University of Glasgow, and today I'm going to be talking to you about biofouling prediction in seaweed aquaculture using eDNA. So there's a few topics already covered, so hopefully there's not too much crossover. So just to give you a little introduction, I started my studies at Glasgow University, where I did an undergrad in freshwater marine biology. I then went on to Stockholm University, where I completed a master's in marine biology. This is where I first got involved with using eDNA during my thesis, looking at phytoplankton seasonality in mangroves. In November last year, I returned to Glasgow, where I've started a PhD, and the early stages of which is what I will be presenting to you today. My project is funded through partnership with the Glasgow University, the Kilcone Milford Trust, and the UK Government of the Research and Innovation under the Biotechnology Biological Research Council Sustainable Aquaculture Partnerships for Innovation project. I've got an excellent team of supervisors supporting me with Dr. Sophie Spatharis, Martin Llewellyn, and Dr. Anna McGregor. And at Kilcone, I'm working with Marnik van Kouter, who's maintaining my experimental sea farm. Just to kind of gloss over this, go to the next slide. Thank you. I can skim this because we all know this anyway. Seaweed industry is growing rapidly across Europe, mostly kind of dominated by Asian producing markets, but we are seeing sort of an increased expansion within sort of Northeast Atlantic. However, there are sort of barriers to this sort of scaling up, and this is because of maybe supply chain issues, processing limitations, storage, that sort of thing. Next slide. In addition to the sort of current logistical constraints on land, there's also problematic factors within the sea itself. The same lovely, cold, nutrient-rich coastline conditions that make the Northeast Atlantic so suitable for commercial seaweed production are also highly favored by harmful biofouling organisms. These epibionts, which are organisms that live on the surface of another, are limited the expansion of seaweed farming in the region. These are species that they drift in as plankton or meroplankton. They then settle on the seaweed as a substrate, where they then establish and quickly proliferate. The bottom left image shows a sort of mat-forming bryozoan, Membranopora membracea, that creates a calcareous crust that can fully cover the seaweed frond. On the right is another seaweed frond with a combined infestation of the circular colony bryozoan, which is like pointed on the left, and this is with a co-infection with a hydrozoan biofouling species, which are more like branch-like structures. Other commonly seen species include groups like crustaceans, such as amphipods, gastropods, mussels, and epiphytic green algae that sort of cling onto the frond. Next slide. So biofouling settlement on both wild and cultivated seaweed causes considerable issues. These include physical damage to the frond by reducing their flexibility, increasing their weight and drag, and this then leads to more breakages and dislodgement. This is especially so during events of high wave energy, like storms. They can disrupt physiological processes by impairing photosynthesis and nutrient exchange. They can block reproductive spore release and can ultimately lead to tissue necrosis. This collectively reduces seaweed biomass yield, and on a commercial level, it leads to significant economic losses through deterioration to the quality, the taste, the overall value of the crop, whilst also increasing processing time and labor costs. Heavily fouled seaweeds are unusable, therefore there's considerably more waste, and there's the added impact of greater allergen risks to humans due to the presence of bivalves, for example. On the image with the arrows, those are small, miteless, edgeless mussel infestations. Next slide, please. So the extent of biofouling coverage is highly seasonal, with farm fouling mainly occurring between spring and summer months, although the timing and the dominant taxa varies significantly depending on the year, the location, and the depth of the cultivation rows. These two plots show biofouling coverage on farm seaweed, with the bottom axis showing time and the y-axis showing frond coverage, which is basically the percentage of the seaweed blade covered with biofouling colonies. The bottom left plot shows a bryozoan seasonal change and shows a rapid increase in the frond coverage in late July, which is just 2 months after the initial establishment was first recorded in May. This is similar on the right plot, showing the hydrozoan coverage over time. And look at this again, we see the significant increase by late July. The blade is almost entirely covered. Due to the impacts of biofouling on the crop and how rapidly the colonies can expand, farmers will harvest the seaweed before the epiphyte biomes typically appear in the water column. Because of this, the farmer's maximum growing season is maybe shortened. And considering that kelp, for example, can grow in 5 centimeters a day, any reduction to this growing season can obviously significantly affect their farmer's yield. It's therefore very important to identify this time window when the biofouling organisms are most likely to be prevalent in the water column, and what are the conditions that optimize their settlement from the water column onto the seaweed surface. If we can identify what drives epibiome dynamics and continue to monitor these conditions, we can hopefully extend the growing season to its fullest and minimize the risks associated with biofouling. We're hoping that this information can inform strategic decisions when selecting future farm locations during initial site planning phases, for example. Next slide. So to develop this approach, we kind of need a better understanding of the sort of biofouling plankton dynamics. In my project, I'm aiming to integrate community data through tracking abundances of biofouling species alongside their surrounding environmental conditions. These predictions will then be validated by comparing them with actual biofouling outcomes observed on farmed kelp. I'll be monitoring key environmental variables, including light, temperature, salinity, hydrodynamics, such as current velocity, wave energy, and nutrient levels. The environmental data will be combined with ongoing sort of epibiome community assessments, which I'll take through eDNA analysis, and then this will be further backed up with microscopic visual surveys. The goal is to identify the strongest early indicators of biofouling and construct an advanced causal model capable of predicting biofouling risks based on these sort of site-specific factors. From this point, we aim to develop an algorithm that can assess the sensor data and then be able to make robust biological predictions to indicate the biofouling risk level of the site. With the sensors that I'm about to talk about, this could be happening like in real time. So next slide, please. This project's pretty unique in that we've got a huge sampling area across a broad latitudinal gradient spanning from Norway all the way down to the south of England. This extensive coverage is going to allow us to investigate biofouling dynamics across a diverse range of conditions, topographies, that sort of thing, making it one of the most spatially comprehensive studies of its kind. In total, we are sampling 11 seaweed farms located in Norway, Sweden, Scotland, and England. These sites were carefully selected to represent a wide range of variables, and this broad spatial coverage will allow us to understand how regional differences influence biofouling risks and the sort of meroplankton dynamics. Sampling's already began back in November, and it follows a structured timeline to capture seasonal dynamics. From last November to March, we'll be doing monthly sampling for a baseline, when the sort of biofilling activity is at its lowest. And from March onward, we'll be doing a bi-monthly sampling to provide a higher resolution data as the biofilling organisms become more prevalent in the water column. This is a combined sort of long-term and large spatial scale. We hope this approach is going to generate a comprehensive sort of understanding of biofilling's appearance, establishment, and succession across both locations and seasons. By combining this sort of spatial and temporal data, we're aiming to sort of refine these predictive models that can help seaweed farms anticipate and mitigate the biofilling risks that are based on their like own site-specific conditions. So next slide. So what am I sampling? We're working with a Norwegian firm, which are specialized in developing sensor buoys. This is soft seaweed. These buoys are going to be deployed to each farm and will continually transmit abiotic data back to us, such as salinity, temperature, and photosynthetically active radiation. We're going to be also measuring current velocity and nutrient content and chlorophyll levels at all the sites. To look at the biofilm communities, similar to what Joanne was saying, we're going to collect plankton samples using this vampire sampler, which is effectively a power drill with a pump attached to it. We'll be passing it through the same eDNA filters I believe he was using. And we've used this in the past, and it's very effective for what we need. From these filters, we will extract the environmental DNA, which is genetic material shed into the water through excretion, shedding, decay. Again, as we heard, this is a really sensitive technique, and it can be really effective at sort of detecting species even at very, very low abundances. DNA from the samples, again, we're going to then amplify using PCR, compare this to a reference database, and from that sort of identify biofilling species, identify which species they are, and estimate their abundances. We're going to be using primers that are targeting bryozoans and hydrozoans. We've chosen these two groups because they're sort of most problematic for the farmers within the region. We're going to be using this eDNA data to identify and measure these abundances of this plankton species. This is sort of the key point that I'm trying to drive across here. These will be sort of quantified using qPCR for sort of relative quantification, and then droplet digital PCR, ddPCR, for sort of the absolute abundance quantification. Combined, this should provide sort of precise tracking of the biofilling dynamics and the sort of severity of infestation within each farm. And this is going to be throughout the entirety of the growing season. The sort of microscopic visual surveys that we're also doing of the plankton will sort of provide visual identification of these species on the actual blades. And this will sort of validate from our findings the sort of what we're seeing in the genetic data as well, and sort of ensure the sort of robustness within our predictive model. Next slide. So just to close, I was saying we're in the early days. We're just sort of getting everything in place. Sampling has begun in some respects, but we're sort of just slowly getting all this stuff together. It's a lot of moving parts. But sort of to summarize, the overall aim of the project, or the key research question, is to determine what are the main environmental drivers for biofilling presences and abundances, and how do these vary seasonally. We intend to answer this by comparing community data collected using eDNA and then comparing this with environmental data across a broad spatial area over the sort of 2 production seasons. As the conditions change over the season, we want to identify this critical threshold that influences the biofilling community dynamics. We want to sort of identify this specific time window that will give farmers the maximum growing season, whilst avoiding any onset from the biofilling impacts. We're hoping that the insights gained from this project will provide farmers with a sort of data-driven strategy to improve productivity, enhance scalability, reduce waste, and just in general overall promoting sustainable growth of their industry. That's some refs as the last slide. And then that's my final slide. Thank you. I hope that was
amazing. Thank you so much. And we'll go right on to the question and discussion section. I'm just going to try to close the presentation so that I can see again. And maybe we can start if there are any questions for Callum straight away, and otherwise I will open the floor for any questions that you might have. I see that we already have some discussions in the chat. That's great. Anything that we can share with the entire group? Nothing. Then I will pull up a helping slide. Let's see here. So we've been talking about a lot of different aspects of monitoring now, both regarding kind of frameworks. There's been some touching on new technology as well, which we're going to talk about more in the later section in the afternoon. But to open up the floor for all of you participants and also for the speakers, I've prepared some guiding questions for our discussion. Sophie and also a lot of the other speakers were touching upon the needs for monitoring frameworks and different kinds of developments, and I see that this has also been talked a little bit about in the chat. But we're also looking at key bottlenecks for effective monitoring and kind of prioritization of what we need to measure. But then I also wanted to kind of lift up that we have a lot of different speakers from different fields. We've heard about seaweed, but also RAS systems and fish. I know we also have some mussel experts in the group. And we are very often kind of limiting our efforts into one of these fields at a time. But maybe there are potentials for synergies across the field, maybe technology or monitoring frameworks that we could kind of share between these different species or just cultivation areas. So does anyone have any input?
I can just confirm that. Sorry, I just went without raising my hand. Just real quick, because seaweed is such a new industry in Europe, not new in Asian countries, for example, but in Europe, yes. So when it comes to carrying capacity, there is stuff already that's going on for bivalve or for finfish, and seaweed is just picking up on this now. So same thing for all of the other frameworks. As seaweed is more of the newer ones, I think there's a lot of more comprehensive frameworks from other aquacultures that we can use and learn, and now integrating this carrying capacity aspect into, for example, the key performance indicator assessments, these sorts of things. I think this should absolutely be done. But yeah, we have the chance now since it's not yet wildly upscaled like crazy, but we're still in a very sustainable way. So I would love to dig into this and to integrate the knowledge from the other aquacultures and use it for our assessments.
Thank you, Sophie. Victor, let's start with you, and then we have Felix.
Yeah, I have a short question. Thank you for the presentation so far. It's been very, very interesting. But I had a question regarding the video monitoring and machine learning. Because we are currently investigating some eDNA analysis in the Baltic to try to catch like fish species that are very difficult to monitor with traditional net surveys, etc., like pike, for example, European pike. But for the machine learning software, the data sets that you use, is that something that you have gathered yourself or learned the machine, so to speak? Or is it are there data sets that are available for different species, or how do you, how do you move forward with that?
I guess it's for my presentation.
Yes.
So no, I conducted my own fieldwork. I went outside and I dropped the BRAV and I filmed myself. So depending on what you are looking for, and what species you are targeting, about like the size and so on, you should adjust the setup accordingly. For example, I filmed downwards, so if there's a big fish, like because my setup was like 40 square centimeters, so if there's a big fish higher than that, you cannot expect to get it with that setup. So you have to adjust your setup according to what you are looking for, either to look downwards or to look straight or in a angle. So yeah, all the data set, it's by me, and also, as I mentioned, it's available to everybody. So ideally, like I think what is in general, like in the biodiversity monitoring, it's the important is the setup doesn't have to be perfect, but if you want to compare different sites, you have to have a standardized methodology. So I guess that's the main thing. Like maybe if you want to build something similar, like and you want to compare, you have to have a standardized methodology, like you will have the same setup filming every time, so you can compare the different sites.
Does it answer your question, Victor?
Yeah, it does. So basically, but just for a quick follow-up, so I completely understand. But basically, you film it and then you identify the species, and then you tell the machine that this is for example, spider king crab, or this is that, and then the machine learns, or.
Yeah, so I didn't have time to explain the whole process. So yeah,
no.
Like eventually, like you film, let's say 5 minutes, and video, it's like a series of frames of pictures, like hundreds, thousands of pictures. So you pick out of a video 5 minutes, it can be, let's say, 5,000 frames. So you pick out of the video, you interact to get 150 frames. And in those 150 frames, you draw boxes where let's say the king crab is located, where the fish is located. So when you show this 50, 70, 100 times for every species, then the model recognize those patterns of pixels. And when you show a different video and it detects those same patterns, then it can say that I'm 72% confident that these pixels are of king crab, for example. But for this, you need to collaborate, I guess, like, for example, I did it with Subsim, where they have access to hypercomputers, because when you want to have big models, you need a lot of resources. So for us, we're using Subsim, the Swedish platform, and they have access to hypercomputers that we can run these models externally when we train the models.
Thank you.
Great. Felix.
Hi everyone. I'm new in the field of monitoring, and I want to learn more about it. So that's why I joined the session today. It's because I'm working on a project to restore the ocean, not with seaweed farming, but with large kelp forests. So, and in order to determine if it's kind of, we know that kelp will regenerate the ocean, but like Sophie said, we also need to know what are the negative and the positive effects, and like Bruce said in the beginning, also kind of what is the cost of deployment, what is the cost for maintenance, and then when you have the social economic values, the negative and the positive, then you have a nice model how you can convince policymakers and other people to deploy these kelp forests. And yeah, so to determine these things, we need to monitor. And so for me, it was very interesting to learn about these the different methodologies. But my question is then, are you all working together? Is there kind of a mapping about who is doing what as a methodology? And is best practices then shared with each other, or is kind of everyone working on a standalone basis? So for me now, if I would like to choose one of the monitoring methods, which should I actually choose? Sophie?
Yeah, Sophie has a good answer to this.
Well, no, it's just a partial answer, because this is exactly the question that I would love that it's already solved, but it's not. But two examples.
But
So
if the wavelength, is that maybe then something which is Submariner is now putting into place?
The
wavelength, Maya, maybe you can answer that.
Wavelink.
Yeah, in the beginning of the presentation, you shared Wavelink. It's a new website from Submariner where I think information about monitoring is put together.
It's
Sorry,
not
I interrupt you, Sophie.
Yeah, no. It's not from Submariner, but I mean, we're engaged in a lot of different initiatives, and there is a lot. Sorry, now I'm hijacking your answer.
Go for it.
But I can just say that there is a lot happening on the monitoring front, and this is also why we wanted to have this this kind of workshop with everybody, because there are a lot of parallel initiatives, and there are a lot of projects where we are developing monitoring frameworks and kind of monitoring schemes for different low traffic aquaculture. And obviously we should do this together and not parallelly in silos, because we want to have a framework that is working optimally for everybody instead of, otherwise we start comparing apples and pears all the time. And it's difficult to get kind of a comprehensive understanding of both of the effects of different kinds of farms, but also in terms of new technology development, like the project that Callum was talking about, or the RAS monitoring. Whenever we kind of switch between these different schemes, it becomes really, really difficult to compare to others. But if you have if you have links or anything to these kinds of platforms, then please share it here and also in Blue Biomatch. And now I hand over to Sophie.
But yeah, just one example, or maybe two examples like from projects. So for example, in the CMAR project, I also did biodiversity assessment, and now learned a lot from that because we made a lot of mistakes in terms of like it was inefficient data collection. We had some eDNA, but we wish we would have done it differently. So we assessed more of the outside world of the ecosystem, well, outside of the seaweed farm, for example. And so now we're in a consortium where we're saying, okay, let's gather all of this info, for example, from the Cool Blue project that was working on biodiversity. Ult Farms was working on biodiversity. Somebody else was working on biodiversity, us. And then in the new project, we sort of start off with gathering info, writing a protocol on testing all of these different systems for their efficiency, for their feasibility, cost effectiveness, and then implementing this in two growth cycles of seaweed, so 2 years, and then really coming up with a recommendation. So it's not funded yet, we're just writing about it, but this would be like the ideal that you start up with gathering all of this information going on right now for biodiversity assessments around seaweed farms. But again, this is just the biodiversity aspect. So this would be one example though, where you combine it all, hopefully, if it's funded. And then another tiny example is that I collaborated with colleagues that work on LCA studies for seaweed value chains. And we said, okay, great, the LCA can integrate a lot of information from like the value chain, the processing and so on. What is the impact? What is the emissions? How much energy does it cost? But not yet to the extent that I study, for example, the negative impacts in the marine ecosystem. So there we collaborated as well in the Algae Probanos project, where we said, okay, let's create a framework that integrates both. And but this is also not yet the one framework that the whole world uses, but it's at least like another example of a monitoring framework where you combine a few things. Now, I listened very closely to what Frederick was presenting this morning on the EU for algae. I was part in a different project, in a different report, but not in the one he was presenting on. And I took note. I was like, this is great. I have to integrate this or make sure that my input is integrated in the next report. So, but it's through these kind of meetings that I try to integrate some of my work as well. Okay, thanks. Many hands up.
Perfect. Asmala, and then Per.
Yeah, thanks. Just a quick comment about the wavelengths for Felix. It was mentioned in my presentation that the wavelengths is a platform developed in the Blue Mission Bonus project, and that's now in very early stages of development. And as it is now, it's focusing on creating links and catalog for projects that are relevant in the Blue Mission Bonus kind of activities. but it's also being piloted internally within the project as a tool for potentially also displaying KPIs and the data behind those KPIs in the future. But that work is ongoing, and nothing fixed is yet to be published.
Great, thank you. Felix, do you have anything to ask back? And otherwise we'll go to Per straight away.
No, I'm fine. Thank you.
You go, Per.
Yeah, thank you. Thank you for some very nice presentations. And Sophie, in your last comment, you had a very short comment on that if it's funded. And I think when we're talking about monitoring framework, when it's finally developed, it will be paid by the primary producers or it will be paid by the authorities. So they definitely want to reduce the running cost of the framework. So I think it's very important to focus on what is needed in relation to economy, husbandry of the production, in relation to the environmental impact, and in relation to you could call it social acceptance. And I think for at least for macroalgae production, social acceptance is not a huge problem now. But if you go for fish aquaculture, for mushroom production, where you have a much more visual impact on the environment, it's much higher impact on the seabed. Social acceptance is very important. So I think in the monitoring, it's important to address how can we actually inform people about what's going on in the water. I mean, there's a lot of warriors on the Facebook that do not like aquaculture. And in the Baltic, you know that the water is damn cold and it's damn wet, and they will not go diving. So we need a way to monitor or communicate what is going on underwater that they accept and understand. Thank you.
Just a tiny follow-up as a clarification, because I completely agree. I think once we have these monitoring frameworks sort of combined and all, then absolutely they will pay for themselves by also the companies. But what I meant is just the research projects to get to this overall combination, because that, you know, like might take some inefficient testing to rule out that's inefficient. We found another way. That's all I meant.
Yeah, thank you.
Sorry. So really, really nice. I have a question to kind of all of you that both have talked and not talked so far. But we've been discussing a lot of different data, and a lot of the data that is mentioned in these projects are very site specific. It's very specific for the farm that it's around, or kind of just the very delimited area that we are focusing on. Are these kinds of monitoring frameworks that we are developing in these projects always useful to actually get the data that we need? Or do you think that there are other kinds of data that we should be focusing on instead? We've heard a lot about eDNA. It's one of these kind of bubbling technologies that we are using in all of the projects. But we also heard some of the kind of limitations with eDNA technology. So maybe some of you eDNA experts can also comment a bit if eDNA is actually something that we should put in use for general monitoring schemes, or if it's good to say something very specific about a specific question or location. Anyone wants to comment? Yeah, Marina. And you're muted.
Hello, I'm a researcher working in with eDNA monitoring and also in the Cool Blue project that Johnny presented. I think eDNA is like for harmonization of methods is really good, because basically you can have a standard protocols that is applied at all sites and agree on using the same methods and the same reference database and analyzing the data in the same by the same bioinformatic pipeline and putting the results in the same place. So from this point of view, it's very broadly applied to any sites, and it can be standardized if just people doing it agree. The only and also eDNA methods, any method has its disadvantages, but if it will be the same for all the monitored sites, so the data will be comparable between the sites. The only big disadvantage I see with eDNA, it's just a little bit expensive methods. So like monitoring many small aquaculture is it's quite expensive, but maybe if it's ordered by authority or if it's done by communities that people like from different places combine few samples, then for each site the cost will go down. Thanks.
Thank you. Barbara.
Yes. I think that's an interesting question. I'm not going to address it from the eDNA perspective, but within this first interview part that we have been having with our partners to initiate this customized development of the tool. One thing that has been really pointed out as being important is know your site. Know your site before anything, before implementing your aquaculture farm, because this is it's a key aspect on the implementation, and to really succeed within good rates of production. So know your site has been pointed out as like a main issue within that. So this is to say that indeed some specificity, some site-specific issues within monitoring need to be present there, because the site conditions, in this case the seaweed tolerances to the conditions that they will be withstanding. This is all very important for the production of the site. So I think that specificity, site-specific data will always have to be something that needs to be accounted for.
Is it going to be possible to get kind of comprehensive monitoring schemes using these technologies in like more broadly in Europe? I'm thinking about, since they are so expensive at the moment.
Can you please repeat the question? I didn't get it.
Yeah, it was mentioned that these technologies like eDNA monitoring, for example, that at the moment they're very expensive. And one of the issues we have with data right now is that it's very fragmented. We have really good data for some places, really long time series or very, very high resolution data. But then in some areas it's completely black. We don't know anything. So do you think that eDNA is it possible to make it a bit more comprehensive in that way, and also use it for regions that don't really have the monitoring frameworks or capacities existing also today? Or is it just going to enhance this kind of gap in data availability between regions?
Well, I think it's, well, of course, the more data we have, always the better. I'm not sure that I'm the right person to really answer this, fully answer this question, but well, we need to start somewhere. Well, and the thing is that data availability, to make data available so that it can be used or even extrapolated or something, this is something that needs to come with larger programs, with larger frameworks, and there we come back maybe to the previous issues. But yes, I think that the more data, the better to understand, like on general on general terms, the conditions that are relevant for this kind of industry, but then we always go back to the site specificity needs that need to be targeted to the applications that are really going on in the specific places.
Marina.
There is a very big genetic monitoring program in Europe, and different initiatives to harmonize them and put the data in the same place. For example, these arms settling panels, the ongoing monitoring program in many sites. So maybe collecting this data, already existing data, can be used as a background data, and then you only need to monitor very close to the site compared to very many places that already been sampled. So that one way. And also with eDNA, is one way is a community composition estimation with metabarcoding and arms of plankton or water DNA. But if you are looking for only some specific species, for example, monitoring the progress of invasive species, or as Callum just told us, looking for biofouling species, and you use a method that target this particular species, and this is much more cheaper and faster. And this is accessible for farmers, for example, if they want to know the presence of, or the peaks of the presence of some species or larvae in the water. That is doable. That is, I think, almost any farmer can afford.
Yeah, it's very interesting. I also know there are a good example on citizen data, and using citizen data for monitoring is the biofouling. I know that in the Baltic Sea around the Swedish coast, they have a system in place for detecting when the exact season of when kind of boat biofouling starts attaching. And it's a completely citizen-based approach. So people just have plates kind of by their boats, and when they start seeing the biofouling, then they record it and send it into kind of a unifying organization. So it is an example of a bit of a different and less technology-heavy approach. But Callum, I'm gonna let you read that.
Just kind of to follow on, obviously I'm a big advocate for eDNA right now, being my PhD. But recently, when we did a pilot study, and there are still limitations, like we did a metabarcoding within a salmon farm and combined that with sort of visual microscopic surveys, and there was distinct overlap where one was detecting one species, the eDNA was not detecting it in the microscope, and the microscope was picking up things that weren't. So we're still getting a lot of differences in this. I feel like eDNA as a single sort of technique, it is getting there, but we're not right there just use it exclusively. And going back to what Johnny's saying, this is all kind of relying on the robustity of the databases that we use for references. I feel like metabarcoding is good for sort of a sort of sweeping statement, but what we're sort of driving at is this species specific. You need to kind of know what you're looking for, have your sort of identifying primers in place to kind of use the technology to find them. So the technology is getting better, and I think as we use it more, it's going to get cheaper. We're hoping to sort of develop things like dipsticks in the future as well, that will be a lot more hands-on and easy to use for farmers like in the field.
Nice. Does anyone have any other questions or discussion points to lift? No.
Maybe I could. I have because I came. I got involved with stakeholders and so on when I want to film the farms and so on. So maybe one input for me, it's why somebody who has a farm to care about the monitoring in general. So for me, maybe it's more maybe there will be some initiatives, if we see that a lot of aquaculture, they help the biodiversity, they enhance it, they attract some species, maybe there will be some benefit for the farmers, for example, maybe, I don't know, maybe not to get extra money, but maybe to pay less taxes, like because it contributes environmentally, it contributes to the sustainable goals of the country. I don't, some sort of initiatives like that the farmers would want to monitor, like they want to contribute to the monitoring system. Otherwise, why they should bother that much unless it's enforced by the government? Maybe that's what I've been thinking when I got involved with such a project. It's like, at the end of the day, why they should bother at all about monitoring systems. Like if there's a way that they could benefit because they contribute to the environment, they contribute that we can conduct some business, but at the same time we don't harm the environment. And if we see that we also enhance the environment, like with species that we attract because of our operation, maybe that would be a good initiative that would push the farmer to seek about monitoring methods, to seek about which method to use, either DNA or monitoring or other methods that are in the field. Maybe I would like to see also other people's opinion about that.
Yeah, really interesting. I'm just to continue on your kind of observation. I mean, there are a lot of monitoring schemes in place already existing for example, water quality monitoring. And there's also a lot of data that if we talk about aquaculture farms that they have to supply to authorities to get permits or to be able to sell their products or whatever. And in general, this is a pretty big bottleneck for the practitioners because it is expensive and it takes a lot of effort. So it would also be very interesting to look at what are the requirements for a farmer, what are the requirements for kind of municipalities or regions in their data collection, and how can we look at how we combine them and combine the efforts to actually get to know more about the environment that we are digging into. Also to create better and more effective monitoring systems. Barbara.
Yes. No, I just would like to say something also on what Zoni mentioned. I think that on top of the environmental concerns, I think for farmers, monitoring is important because it will actually give them information on their production. Because in the end, we also need to see this as a business. So the monitoring will provide them information that is crucial for them to know how to act, how to intervene, so that they get a better profit from it. But of course, there is the thing that's monitoring as costs, and I think someone else has also mentioned that these monitoring systems sometimes they're quite costly, and that's indeed getting alternatives that or funding systems that allow them to implement these monitoring systems, not only for their own production efficiency, but for the monitoring of environmental impacts is something that will maybe benefit to everyone. And maybe now stepping into the bottlenecks on this effective monitoring, maybe from the technical point of view, something that we have been also trying to address, and something that it goes related to Kaelin's presentation earlier. So biofouling that was mentioned to be very to impact on the crop itself. It's also something that impacts the monitoring systems. So it needs to be I don't know if it's a bottleneck, but it's certainly a challenge to understand the harmful things that can affect the algae. They also impact the monitoring systems, and is something that needs to be overcome so that we can understand how to prevent and better understand when these conditions are going to come, because this has costs in all senses. So on the crop, on the systems, on everything. So the environmental conditions it's something that it's exposed to harsh conditions. So in any ways, everything is going to be affected, and it's a challenge to understand them and try to overcome the effects that it has not only for the crop but also for the monitoring systems.
We have 3 more minutes on this first part of our big monitoring day. I just wanted to throw out a last question to all of the speakers, because you are, I mean, the reason why we do monitoring is to collect data so that we can actually say something about the environment. What do we do with this data to make it available? Are you sharing the data in the existing platforms that we have, or how do we enable other people to actually use the monitoring data that we do collect now?
Well, I can go. For us, we have 11 farms that are all involved. They're all sending us samples back every month, every few months, and we're it's going to be a combined. We're getting every information that we get, we're giving back to them. And like I said in my talk, we're going to hopefully develop this sort of predictive tool that we can sort of roll out to not just their farms, but obviously any farms that want it. And what we're hoping is that these sort of simple abiotic factors that we measure, that can sort of just tell us the sort of biofouling risk. And we hope this sort of combined data will be easily available to sort of the farmers like every day, so sort of mitigate the sort of risk that we're getting. We feel that the most profitable, beneficial for all people is sort of combined efforts. Things like this, talking with like each other, like problems that other farms have been the sort of the most informative for us in sort of developing this project. I think cross-party collaboration has been sort of the best thing for this whole industry to sort of like upregulate.
Nice. Anyone else?
From my side, the framework that I presented, it's about to be published. It's just, it came back with review, so I'm expecting another couple of weeks until it's actually published. And the data that we collected in the CMARC project, that will also be published sort of mid of the year. It's right now being written into report and then absolutely afterwards published and available. Yeah.
Well, from us, we haven't actually started collecting any data, but I think that well, the making this data available will depend also on the farmers that are that will be with whom we will be testing our system, and of course the type of funding that helps us to get this to this testing. So this is something that it's not yet decided on how the data will be available for everyone.
And in my case, that I got like species detections, like the model, it's on Zenodo, where it's a place where a lot of people upload their datasets or models. But also we're planning soon also to upload those species detections also in GBIF, like people can also use them later on for when they want to see like what species were found where, and they can contribute to the whole system, like when they want to make like species distribution models and habitat suitability. So that's at least for us when it comes at least to species detections. And I can also post in the chat in case somebody would be interested.
Yes, please do so. Great, everybody. Thank you so much to our speakers. Really, really interesting and really nice discussions as well at the end. And we will now take a bit of a break. So we'll go for lunch and then we'll be back at 1. And then we're going to focus a little bit more on the specific technologies and technology developments for monitoring uses. And please register in Blue Biomatch, everybody, and you can continue the discussions there. Or if you want to ask anything to any one of our working group experts, then that's the place to be. So I wish you all a nice lunch, and I'll see you in about an hour. Okay, I think we're gonna get started. I think some people are gonna be joining us right now. So I'm just gonna say that this is a recorded webinar. So if you don't want to be seen, then turn off your video. And also some house rules that if you're not talking, which you shouldn't be doing if you're not a speaker, please mute yourself and keep yourself muted. If you have any questions, please post them in the chat or raise your hand before. And with that being said, I think we're gonna jump straight into it and leave the floor to Per, who's gonna talk a bit about drone monitoring in Baltic Muppets. Per, are you here? Are you ready?
Yeah, can you hear me loud and clearly?
Amazing, and we can see your screen as well, so you can.
I just start. So, yes, I'll talk a little bit about drone monitoring in mussel farms. It's a part of the Baltic Muppets project financed by EU. Why should you produce mussels in the Baltic? You should, of course, produce mussel because it's good food or feed, and at the same time you improve the water quality in the Baltic. You extract nutrients, you increase the transparency of the water. So there's a lot of benefits from producing mussels. Yes, we have 3 pilots productions in the project. In the Limfjords, we have a submerged system. 120 submerged production units are established. I will come back to that. Then in the east coast of Sweden, in Västervik, they have another type of submerged mussel production. And in Kiel, they have a long line system where the main objectives is to protect the system from eider ducks by a fenced system. But for the Swedish and the Danish system, when you submerge a mussel farm, it's actually difficult to harvest the system, but it's also difficult to monitor what is going on. What is the biomass? Do we have any problems with invertebrates eating the mussels, pests, so on? So we need to develop some new monitoring systems to check what's going on below the water surface. If you take a look at the Danish partners, when we are 2 Danish partners working with a pilot in Limfjord, it's a Blue Research where I'm sitting. It's a consultancy company that we are specialized in developing different types of aquaculture, including mussel farming, but we are also working with marine ecosystem restoration. And in Baltic Muppets, we are working on a system to monitor. The other partner is Vitrup Seafood. They are a primary producer. They produce mussels, but they also work with habitat restoration, and some of them, the mussels are used for biogenic reefs. And in the Baltic Muppets, they established 120 submerged mussel farm units. I can show you a little bit more about that. That's a smart farm system. Each unit is constructed by a 120 meter long PE pipe, and below the pipe, it's a closed pipe. There is a net, and on the net, a lot of concrete blocks are attached to submerge the system. So this is some pictures of when the farm was established. And in the lower corner, you can see how many mussels we actually had after, I think it's after 3 or 4 months. Yes, what we are using under one drone for the monitoring. It's a standard drone, and on the drone there is attached a sonar, so we can actually log the distance between the drone and the mussel culture. It means that we get some standardized pictures that can be used, and we use the pictures at 3 different ways. We produce photogrammetry with 3D models that are used to estimate the biomass, and we use AI to count individuals. I'll come back to that too. And furthermore, we use a 360 degrees camera for demonstrate for that's more for communication. If you go to the this is a picture. This is actually a 3D picture. I can show you. You can if you search on Sketchfab, you can search on Limfjord Muslinge. Then you get this model, and it's actually a 3D model produced from about 5,000 pictures taken from a drone, and you can see it's actually a really detailed. It's rather detailed models where you can see starfish. You can see almost individual mussels. Sorry. So this type of information is really good to communicate what is going on below the surface, the water surface. Yes, if we switch to how to produce the 3D model, then with the drone, we take a lot of pictures of a small area, and then we use a program. It's called 3DF Zephyr. It's a program that relate all the pictures to each other and then produce a 3D model. Here you can see a model produced from about 50 pictures. It's a video from a screen dump, and you can see there is a lot of details. And next step is actually that we can define a level, for example, the net, and then we can estimate. There's a function where we can estimate the volumes of the mussels and of course the sea starfish, and this estimate can have to be correlated to the harvested amount of mussels, the harvested volume of mussels. So by this system, you can you can actually estimate the volume and the biomass of mussels. Yes. Then we switch. I mean, we had the presentation of Cool Blue, where you are the YOLO 8 model was presented, and we are using the same. We use it on a Google Colab and use the YOLO 8 model to making a program where we can actually analyze individual pictures of the mussel culture, and we can use this analyze to estimate the number and the size of different animals. It's not that difficult to setting up because you have the Gemini. It's an AI system that actually help you to formulate the model. The first step is that you start to train the model. You have a system where you make an annotation of a lot of pictures, where you manually set and identify where do we have a starfish, and then you set a box around each starfish, and for each picture a small file is produced that describe each box for each starfish. And then you can train the model, and you can, I mean, this information, I'll not go in details, but it describes how you train the model with let's say 50 to 150 rounds, where you train again and again, and then you reduce the loss for each run, and then you can actually use the model to identify where do we have starfish on pictures taken on the mussel culture. And I just show you two pictures from the model is not trained to perfection, but it's just indicates that you can use the model to identify starfish. And furthermore, I have prepared the program so you get a list of number of counts on each picture. So in that way, when the model is perfect, you can analyze a lot of pictures taken by the drone. The last technology I will shortly present is the 360-degree camera. It's a camera we put on top of the drone, and you can use the 360 degrees camera to set up a presentation to inform, to communicate to people how it is to dive along a mussel farm without being wet. If you take your iPhone and use the QR code, then you will get into a VR video, and you can move your camera, your iPhone around, and then you will see the mussel farm from different directions, angles. And if you look backwards, you can see the drone that actually working next to the mussel farm. So I think that's. Thank you for listening to this presentation. Thank you.
Thank you, Per. Does anyone have any questions for Per? We have some minutes if anyone has anything now. Yes, Anda.
Yeah, thank you, Per, for this marvelous presentation. I was really amazed about this work, and I just wanted to ask whether you assume, when estimating the biomass, that are all mussels alive, or how do you deal with that?
Yeah, when it's farmed mussels, normally there will be very few dead mussels. But if you take a mussel culture from the seabeds, then you will have more dead mussels. But normally dead mussels is not a problem on farm mussels, on long lines or on smart farms.
Great, thank you, Anda. Anyone else? Yeah, Sonny.
Hi, Per. Thank you for the very cool project. I want to ask, have you run the model on unseen footage, or it's run on the trained video?
It's not a video. It's on pictures from a GoPro camera.
Ah, okay. I thought like you filmed and then you run it as well. You run the model on the video.
But I think in order to reduce the amount of data that you can do the same on a video.
Okay.
But anyway, I had the pictures from the 3D model, so I use the same pictures.
Okay, very cool.
And sorry if I was telling that it was simple to make the model, but I think it's rather simple to set up the model when you use the IE helping functions and stuff like that.
I mean, essentially, maybe it could work also on the video, since if you're filming on the same way, and it will be the same setup, kind of. So maybe if you run it on a video, maybe it could track something, I guess.
Yeah. I mean, you just need to make a small modification of the program, and then you can modify it to using the video. And then, but I mean, as long as we are looking at starfish, they are not moving around. But it could be interesting to see how, for example, small fish, they use the muscle culture, how the height between the muscles and stuff like that. And there, analyzing the video could give you much more information about how they are moving around on the muscle form, on the muscle culture. Yeah.
Great. Do we have any more questions? Otherwise, we will hand over to our next speaker straight away. Then I would like Anak to, are you there?
I'm here. My name is Anaik.
Perfect. From Ultrafarms about marine monitoring offshore technology. So can you maybe try to share your presentation?
Yes, I will do that. Is it working all right?
Looks, yes, that's perfect. Thank you. And the floor is yours.
Thank you. So hello, everyone. My name is Anaik van Gerven. I work at Orbins, so that's the Royal Belgian Institute of Natural Sciences in Belgium, thus. So here I will present today the monitoring strategy that we've made for offshore aquaculture as part of the Ultrafarms project. See if that works. Yeah, okay. So Ultrafarms is a Horizon 22 project about ocean multi-use. So we're specifically trying to combine offshore wind farms and low-traffic aquaculture. The aim of the project is to demonstrate profitable, sustainable, and eco-friendly low-traffic aquaculture that is shared with offshore activities, such as wind farms. Okay, so I will not read all the lines here, but the purpose of the project is to move forwards in terms of technology because so far it has been very conceptual. We had a previous project about this type of multi-use before, and we were really trying to advance on very practical matters like technologies, administrative issues, feasibility of combining these two activities at sea. For those of you who are not familiar with multi-use, what is ocean multi-use? It's basically combining two or more activities in a single area. It can have diverse forms. It can, depending on how much is shared. So you can share space, time, infrastructures, operation, etc. Some of that, or only one of these aspects. And examples of multi-use are offshore wind farms with aquaculture, offshore wind farms and solar energy production, tourism and aquaculture, tourism and active restoration, etc. So why should we do ocean multi-use? Because we have crowded seas, so we want to resolve conflicts over space. It also allows to make some space for smaller, less powerful sectors, such as low-traffic aquaculture, and it could generate environmental, economic, and social synergies. In the project, we have 6 pilots in Denmark, Germany, Belgium, and the Netherlands that try to combine renewable energy production with mussel, oysters, and seaweed aquaculture, and also in some cases with nature restoration. We are all in the North and Baltic Sea. So, as part of this project, we try to produce a monitoring strategy for offshore low-traffic aquaculture, because low-traffic aquaculture in wind farms usually means offshore. There are exceptions, of course, but it's very likely that it's going to be further to shore than what is used to for aquaculture. And of course, working offshore comes with its own set of challenges. So here we've designed a sort of ideal monitoring strategy. It's not always possible to apply it in every case because, well, it's what we want to aim for. And it consists of 4 Excel tables that are divided as follows. The main structure is that we will have a baseline data collection that will allow to assess the site suitability and provide a baseline for future comparison. And then a post-decision monitoring. So that's going to be the monitoring during the lifetime of the project. And this monitoring is further divided into operation, product, and environmental monitoring. First, the baseline data collection, it's to be done before anything is installed. So again, this is an ideal scenario. It's not always possible. Often the wind farm will be there already. So, okay, we work with what we have. But this baseline data collection, it can serve to choose the right location, or if the location is already set by the wind farm, to assess if this location is suitable for aquaculture in general, or for a specific species, for example. And often data for the baseline data collection will already exist from public data, or it would have been collected beforehand. For example, if you have a wind farm, often like the ground, like a lot of data is already available for the area. And so, as I said, we have two main purposes for collecting the baseline data, and that's to assess the site suitability and then to produce a baseline for future comparison for impact assessment. So, very pragmatically, this is what the monitoring strategy looks like. So we have we looked at the different variables that you could monitor, why you should monitor it, the indicators for these variables, and then also in the countries of our pilots, if there is a legal obligation to monitor this. And then we suggest some methods. So the methods are not exhaustive because there are a lot of different technologies out there, and we couldn't list them all, but these are at least ideas to begin with. So for the baseline data collection, you want to look at your seabeds. Why? Because you're going to have to anchor stuff for low trophic aquaculture. And you might also want to look at your carbon stock for further future impact assessments. Then you want to look at how the ocean moves. So swell, wave, current, tide, wind also, because this will impact the infrastructure that you will have to design for low trophic aquaculture offshore. Then we want to look at the water's characteristics, such as its content in nutrient, plankton, its pH, carbon dioxide content, oxygen, contaminants, etc. This is going to be important for the species that you raise, for the organisms that you raise. And then you want to look at the biodiversity in the area, because this will provide a comparison point for impact assessment later on in the project. So you want to look at your benthic community, your fish, your mammals, your birds. This could be more detailed or extended depending on where the project happens. Then the post-decision monitoring. So this refers to what is to be monitored during the lifetime of the activity. We have divided this into three main aspects to be monitored. We want to monitor the operation itself, the product that's cultivated, and then the impact of the project on the environment. So when regarding operation, you want to look at your yield and your infrastructure. Your yield, you want to monitor seaweed bivalve growth and diseases. Why? Because, well, you want your project to be economically profitable. So if you have a disease or an issue, you should know it as soon as possible. And then your infrastructure, long lines, buoys, and other structures underwater. This is for safety and damage control. Because when you work in a multi-use context, whatever you could lose at sea is it could damage the wind turbines, and that's a very high cost to repair. Then looking at your products, of course, if you're raising organisms to be consumed, you have to look at your food quality and food safety. So I will not go through all the lines here, but you get the concept. So you want your products to be good enough to be sold, and you also want it to be safe, of course. And finally, in the post-decision monitoring, we have the environmental impact monitoring. So this can be crossed with the baseline data collection afterwards. So you want to look at your water quality, how you impact your nutrient and your plankton with the organism that you put that you raise. You want to look at your soil. Are you stocking carbon or not? Because there is some hopes for at least seaweed aquaculture to store some carbon into the ground. And then you want to look at your sedimentology. And finally, to look again at your biodiversity during the lifetime of the project and maybe after the lifetime of the project, compared with the beginning with the baseline data collection, to ensure that you have minimally impacted your benthic community, your fish, your mammals, your birds, and maybe also if you had a project of combining the aquaculture with habitat restoration, if this had an actual impact on what you wanted to improve. So in the document, you can see in the tables that we have set the legal obligations for the countries that we have a pilot in, where there is a little star. It means that there are conditional legal obligations. So sometimes it depends where it is, the size of your farm and so on. So this is all detailed in the full report that presents this monitoring strategy. The full document is available through this link. I don't know if the presentations will be shared afterwards, but otherwise you can always email or go on the website of Ultfarms. The report is there. And this includes a description of the monitoring strategy, so what to monitor, why and how, and then also a description of what is legally obliged in the countries of the pilots. This was written in spring 2023, that's almost 2 years ago. I don't think it has changed much yet, but I assume that the legal obligations will change in the coming years at some point. All right, are there any questions about this?
Thank you so much. Yeah, do we have any questions? We have some time. Very, very interesting to get some multi-use perspective on the monitoring as well. No questions. I have a question. How would you say that the monitoring in multi-use contexts is like? What are the main differences compared to more conventional aquaculture? You mentioned a couple in your presentation, but I'm just wondering.
The biggest difference is that it's going to be further to shore. So working offshore, well, you have different constraints, you have different things to monitor. You're going to have to use also very different techniques because you want to use a remote and automated systems as much as possible to reduce how often you have to go at sea. Also in this season, it's a big issue actually in the projects like in Ultfarms and before that we had United. Working offshore is very costly. It's not easy even to find if you don't have your own vessel. It's not easy to find a vessel that will take you when you want it. Also, the weather often cancels whatever you're trying to do. So working with remote and automated systems is very important in this context, working offshore. And I think the fact that it's combined with wind farms, as I mentioned, it makes it even more important not to lose anything, because we've also had the case in one of our projects that a pilot had some structures getting detached. And so it was actually recovered in time, but if you let it drift too much, it goes in a turbine, and repairing the turbine is very, very costly. So then you have big insurance issues. So, yeah.
I guess that's a challenge as well if you want to do things remotely, but also reduce the loss, because usually it's maybe a little bit connected. It's easier to do something that you don't have control over at the moment.
Yeah, that's true. Well, multi-use comes with a lot of challenges.
Yeah. Perfect. Does anyone else have any questions for Annick? No. Then we're going to hand over to our last speaker for today, which is going to be not on low traffic, as I understand it, but on fish farming. And we have Oxygard here, Mette.
Yes, I will just try to share my screen.
Yeah. It looks good.
Okay, I'll do this. Is it still good?
Looks great.
Okay, Thank you. And I'm so sorry I don't have a camera, everyone. It's just the setup I have today has no camera. So my name is Mette, and I work as a senior scientist at Oxygard. And today I will be talking about the state of the art of the monitoring and control, primarily in RAS. With the point that even though we call it state of the art, it's possibly outdated. And I will talk about how we at Oxygard address this and how we work to find solutions. So much of what I will be saying in my talk, others have been saying during the day, but I guess that's like one of the downside of being the last speaker. So I hope it doesn't get too boring. First, a little background on Oxygard, just if we have listeners here who haven't heard about us. Oxygard specializes in developing advanced solutions for monitoring and control for agriculture. We started all the way back in 1987. So today we have more than 38 years of experience. And that means that we are among like a few number of companies in the field with such a long history. And this has actually allowed us to gain a deep insight into the challenges and advancement in agriculture technology. We consider ourselves market leaders within several categories of equipment. We have valued customers in more than 120 countries and a strong network of distributors in more than 60 countries around the globe. Oxygard is also a very special company in several ways. Especially compared to other equipment producers. First of all, we have our entire business, including the production unit, placed in Denmark. And this is special because the production expenses, including salaries for our employees, are among the highest in the world. So we could actually halve the price of our production by moving it abroad. But we believe that the social aspects count as well, and we want our employees to have a good salary and a good work-life balance, and also to keep the production in Denmark. We have a very fast response time when we bump into production errors and stuff like that. Another key aspect of our work at Oxygard is our commitment to research and innovation. Even though we are an equipment provider, more than 30% of our staff are dedicated to research and development, and this includes 6 employees holding a scientific PhD. And actually, within the next month, a 7th PhD will begin, and he will focus on application of artificial intelligence to analyze visual and acoustic signals in agriculture system. So innovation is very central to our business strategy to stay at the forefront of the agriculture technology development. We also actively participate in a broad array of research and innovation projects. And over the last 8 years, we have engaged in more than 16 collaborative projects with 39 partners in 24 countries. And this global network enables us to contribute and also to benefit from the more like collective advancements in agriculture science and technology. So the scope of this presentation will be to outline the current capabilities and limitations of monitoring and control technologies in RAS. But I will primarily focus on the future opportunities and the more like scientific and technical challenges associated with achieving this comprehensive integration of monitoring and control. So when we talk about monitoring and control, like 10 years ago, we were mainly focused on the water matrix parameters, but today that's just part of it. We have like the water quality is just one area of monitoring. Then we also today monitor like the more operational logistics, such as tracking the feed use, the inventory, workflows, personnel activity, and stuff like that. And also we're beginning to monitor stuff like fish behavior using a camera and hydrophones to access stress responses and other indicators that could enable improvements somewhere. And then Also a new thing, we also have these ESG goals. Many of you must have heard of them or even be under them by now, that like aims at the responsible production across all parameters. So this is also something that will be included in monitoring. I will quickly go through like what we could call the state-of-the-art monitoring and control system today. So we can define kind of a technology ladder, and in one end we have facilities who need just a low degree of technology, perhaps only like a handheld oxygen meter would enable them to optimize. And on the top of the technology ladder, we would then have the inland RAS facility that needs an advanced setup of monitoring and control equipment and an equally advanced water treatment system to run it. Today, we offer a broad range of monitoring equipment covering nearly every critical parameter you have in the water matrix. And most, if not all, of our RAS customers use our Commander Pacific system. And this is what I have shown on this slide. It's like built up around a central unit, and all the different sensors you have in your tanks will feed into this central unit. And from this central unit, you can then put in some levels of control, like you can set the threshold for values for water parameters by your own choice, like oxygen, pH, or temperature, or whatever you choose. And you can have the system give you an alarm if the values are outside your threshold values. You can also use the central unit to access tank-specific alerts in case of deviations. And then you can implement some automated regulations of different variables, like if you have oxygen dropping below a certain level, you can turn on the aerating system or add oxygen. But it's still today, it's more like on-off switches you can control in this way. The key advantage of this system I show here is that we made it like a modular design, allowing it to be expanded and customized. So even a small fish farmer could use the system, and also the biggest in the world use it. And you can build it much like a Lego structure to accommodate additional functionalities you would have. So we have several additional add-on modules with different functionalities. The system can also be accessed from a computer, and the different control possibilities can be accessed from anywhere, and you can get your alarms even on your holiday anytime you want and from anywhere. So there's a great level of safety in these systems as well. And then to conclude the state of the art today, on top of a farm management and control system, you would most likely have a digital platform on top of this. And we have made our own digital platform, Cobalia. And as I show in this figure here, Cobalia is like combining the top, which is a more farm-specific data. You can feed data from the Pacific system. I don't know if I can show my cursor here. Yeah, here you have the Pacific, and the Pacific receives data from all the different areas of the production, and you feed that into this your computer where you have your digital system, and you can also use your phone and some tech cards to transfer like tank-specific data of your choice to the system. And then the digital platform adds a new layer of both a new layer of monitoring, but also a new level of functionality. So you can have the system automatically order feed. You can choose whether or not to share your data. You can have the system inform the veterinarian that he needs to come. And then in the bottom here, we have all kinds of functionalities that are beneficial for the fish farmer to make the platform interesting for them. So we have a new level of warning and alarms based on other stuff, and you can have the system automatically produce facility reports. You can have mortality trend. You can have it do graphs and bars and reports and all kinds of stuff. so with the introduction of the digital systems, we also introduce to some degree AI, artificial intelligence, and this is kind of where I want my talk to go, because with the AI we're facing a new reality, both in fish farming, but definitely also in monitoring and control. So currently monitoring and control systems operate like on an individual parameter basis. So for example, we continuously measure dissolved oxygen, and then when oxygen falls outside the preferred range, the system automatically activates aeration or oxygen injection. And then once the optimal levels are restored, the system shuts down the oxygenation process again. But this is highly efficient, but this is also what we call a linear control or a reactive control. By introducing AI, we can take it a step further. So rather than reacting to threshold breaches, AI can analyze all the parameters influencing the oxygen fluctuations, and then we can have the AI predict whether or not there will be an oxygen depletion before it occurs, and then we can proactively regulate the oxygenation with much higher precision, reducing the operational cost and increasing the system stability. So this shift from reactive to predictive control is like one of the most exciting things that are happening in aquaculture monitoring and control. And this is one of our main focus in our research and development at Oxigard. So to be able to actually conduct this predictive nonlinear control, we need a lot of data. We currently concluded a major project called IntelliRAS in collaboration with Luke in Finland and the Norwegian Veterinary Institute and the University of Aarhus and a number of Danish primary producers. And through this project, we have evaluated the data availability in RAS facilities, and we have assessed whether or not data-driven precision fish farming is feasible or not within the current industry practices. So our finding highlights a significant gap in the data collection. We don't nearly have enough data to drive neither AI or precision fish farming. And most fish farmers, what we have learned, only continuously measure oxygen and temperature. Then some of them collects CO2 on a continuous basis, but this is not a standard practice. And all the other key water quality parameters are recorded only like discrete data points rather than this continuous time series data that we actually need to train the artificial intelligence. Whoops. Yeah, I skipped one here. So through this study, we identified several key barriers that needs to be addressed before AI can be effectively integrated into aquaculture and into monitoring and control. And we will actually send out a white paper later this year, so you can look out for that. And for this presentation, I will just mention the top 3, and all of them have been mentioned today already. But the first and biggest is like the lack of sufficient data, because farmers collect only the data necessary for the daily operations. And this continuous high resolution data collection, it's expensive. And when the fish farmer don't have any immediate operational benefits, they simply don't have the incentive to invest in it because it's expensive. Then we have another big problem is that there's no standards in how to collect data, in which units to use, and how there's no protocols for this. And this lack of consistency makes large-scale AI training very, very difficult. And a last thing is that we have this very limited capacity to store and transfer big data, and we get big data when we, for example, use cameras to track behavior. So we don't have a good solution right now how to store and transfer this data. So in response to these challenges, we have developed the digital system I told you about before, Cobalia, and Cobalia is designed for a seamless AI integration. So it's very cheap because we want every fish farmer in the world to be able to afford to join. It's a platform that are controlled by the fish farmers. They own their own data. So this is kind of like a tool we want to be accessible for everyone that would benefit from improving the monitoring and control regimes. Our current focus now is to improve the data accessibility and to, through Cobalia, create some real value for the fish farmer in data sampling, so that this platform will enable continuous monitoring of all the key water parameters, both inside and around the farm, while at the same time providing the fish farmers with benefits that will give him a return of investment. So actually we try to make this this platform so attractive to the fish farmer, and that to give him so many benefits that the adding on with new data will be a minor thing for him. So kind of a trade-off for more direct benefits. Here's just the data we would like. It's really a lot, and probably less could do it. I just like listed from the top of my head what we really would like to get, and as you can see, it's quite a lot. And for all the water matrix parameters I have here, you have to keep in mind that the fish farmer will have to buy the probes, add them to the tanks, add them to a monitoring system. So it's very, very expensive. And I also added here how we would also like to have monitoring on the stuff that will feed into the greenhouse gas data, the workflow, the logistics, and also on the other end of the production, health status and meat quality. So the AI in the long term can correlate all these things together and point back to how to improve the subject of choice through within the production. So with the platform like Cobalia, we will get some of the way, but not all the way. In order to make the AI attractive enough, we need to train it before actually asking our customers to buy it. And we try to do some of this through our many developmental projects. And we intend to apply for a larger EU project this year or next year, where we want to address the implementation of real AI in agriculture, as well as establish a platform suitable for the ESG application, and also some of our new technologies we're working on, such as identifying behavior using cameras and hydrophones. So I would actually like to reach out for partners interested in these types of projects. So I just want to say, feel free to get in touch if you want to be involved in this application. And then I just want to say that all this AI is a major milestone in the digital transformation of agriculture. And in the coming years, our goal at Oxigard will be to bridge the gap between monitoring and true AI-driven control. And that was all I had today. Thank you so much for listening.
Great, Mette. Thank you so much. And nice with a real call for action and collaboration. It's really, really interesting to hear a bit about the kind of high technology developments as well. And it sounds like there are a lot of overlaps or potential synergies together with other presenters. Does anyone have any questions directly to Mette now? And otherwise we open up for some general questions or general discussion. Yeah, Michaela.
Thank you. Hi, Mette. Just a quick question. On your slide there where you listed the parameters that you would like to be able to measure, you listed ammonia and then nitrite and nitrate in brackets. Do you currently have a solution for nitrite and nitrate monitoring, or?
No, we don't. We don't. Not for online monitoring or measuring, but we're working on a secret sensor. But we don't at the moment. No, we will have to take a water sample and use a kit to do it. Yeah.
Perfect. Okay, thank you so much.
Anything else? Yes, Per.
Yeah, thank you, Mette, for a very nice presentation. It's really interesting that you produce a digital twin that you actually send into the future, so you know what will happen with a physical twin, you could say. But I mean, you list a lot of parameters. A lot of these parameters, they will be correlated in a way. So I mean, maybe it's important to focus on what is actually the key parameters that are needed, and that you can produce models from these key parameters.
I totally agree. I also did that slide to kind of create a shock wave because it's true, many of the data we can actually get them from having access to other data and stuff like that. So we are aware of that. I just like to put it all out there. This is what we want.
Yeah. But it's I mean, the fish behavior is really interesting, because maybe you can get an early warning from the behavior from the fish before you get it from some of the other sensors, or from the sensors.
Yeah, we have some pretty interesting results on the behavior. Actually, we try to focus on monitoring behavior around feeding, just to train the kind of like you do with your model, I guess. So we annotate a lot of video, and then we can, to begin with, just look at the activity, but also how much they snap in the water or jump or whatever, and then we can begin to correlate that to behavior, and it's very interesting. It's a good time to be in the business. It's so interesting.
Thank you.
Anyone else has any questions for Mette? No. Then I think we can open up kind of a general discussion or any general questions to any of our presenters. Also, feel free to, you can address anyone from the previous session as well before lunch, when we're talking a bit more about monitoring schemes. Hopefully some of them are still here. It's been a long day. Yeah, just adding on the topic about technology use and technology development. Does anyone have any immediate questions or discussion points that they want to bring up? Otherwise, we have our kind of guiding slides here. Nothing at all. I'm thinking a little bit about this, the developing technologies that are kind of emerging now. And we had Mette talking about kind of AI and also remote monitoring, which was one of the needs from old farms for the multi-use monitoring of multi-use cultivations. Do you think that there are any collaboration opportunities here, or are the monitoring technologies for kind of RAS systems so specific that they can't really be applied in the outdoor setting? I think that's for either Mette or
Hi, I'm so sorry. I was just at the. I was just in the toilet.
No worries.
So you have to ask again if I'm to answer.
Oh,
Sorry.
yeah, no worries. No, I was just asking that because so Anna was talking about the needs for from multi-use cultivation monitoring for kind of remote, either remote sensing or kind of remote controlling of any monitoring equipment. And you also described your technologies with kind of IoT solutions that could be done remotely as well. And my question was if the technology that you are developing or that is currently developed for RAS, if it's too maybe fragile or specific for kind of the RAS setting, or if this could also be developed in collaboration with the multi-use application.
I don't really see this as a threshold actually or bottleneck because we do have equipment for on-sea monitoring as well. It's not where we see the challenges.
Per, you have a comment?
Yeah, I think one of the technology platform that will be common for whatever it's RAS, if it's seaweed, it's mussels, it's biodiversity. That's actually the AI analysis of pictures and videos. I mean, we have several presentations now, and it seems like this type of technology can be used in a lot of different applications. And it's at the same time it's become much more not easy, but it's become much more easy to set up a system that is actually working. It's easy to program because there is so many helping functions helping you to formulate the models and stuff like that. So I think in the next 5 or 10 years, this is definitely a growing area that should have a lot of focus.
Michaela.
Yeah, just a quick question. Like I said, just to reiterate, I'm no expert in RAS specifically, but I can speak on the technology side. And in terms of aquamonitrics, I don't know if Meta was there for my presentation earlier. I probably fit in more with the after lunch group. So sorry about that confusion. But yeah, in terms of what Per was saying about being able to, it's not so specific to RAS only, and kind of that these technologies, I think, Maya, did you mention like the word fragile? We have worked as part of different like Horizon 2020 projects as well with partners in Greece that are doing like algae applications. We have partners in Spain who aquaponic facilities and hydroponic facilities. So there really is, the applications are endless. And I know I talked at length about the salinity benefit of the aquamonitrics analyzers too. So if there's anyone present that kind of has an interest in that, obviously that's a big aid in the marine monitoring as well. So yeah, just kind of to add to that a little bit.
Thank you. But you also mentioned that you don't think that the kind of technology is the main bottleneck. What would you say is the main bottleneck for you?
The main bottleneck for us is to access the data we need to train an AI, because it's very, very expensive to collect the data. You need to buy an expensive sensor. You need a system to access the data. You need to transfer it digitally. We have challenges related to the expense of the system, but also just the transfer of the data. We have done video monitoring of salmons, and it's a tremendous amount of data we need to store and then transfer. And we are working on some solutions where you do part of the data handling within the camera, but we are not there yet. So I would say that is the main challenge. when it comes to the technology for the specific sensors and probes and systems, it's more easy to do it. Like I said, we are even trying to do sensors that can do online measurements of nitrate. So I don't think that that's a bottleneck, even though we still lack some of the sensors. So it's more, we are on next level now. So we have to consider especially video, AI, and integration.
And maybe you can help there, Per, with your remote camera. Does anyone else have any questions? No, then I will continue a little bit because we're talking about the costs of monitoring, and here we're also talking about the cost of the kind of data and the technology for monitoring. Something that is discussed a lot in kind of innovation forums, and also when we're talking about circular economy, is the sharing of infrastructure. And here we're talking also about multi-use again, sharing kind of space. So how could we facilitate more of a sharing economy when it comes to monitoring, to both share the technology we need for monitoring, but also the data that we need to develop these models? Do you think it's possible, or? Mette?
Kind of related to what you just asked, we believe that when you talk about how to share the data, we have a number of digital platforms out there where the provider of the platform will actually own the data. And then they will use the data for the benefit of their company or whatever they need to use the data for. We consider that a problem because what we have done is to say, when you use our digital platform, you own the data. It might seem counterintuitive because we actually need the data, but we know that the fish farmers, they will use that data. Their goal is to produce the best product for the lowest price. So this is also what we want. So if we allow them to control the data, like the fish farmers themselves, we strongly believe that the development will go in the right direction. So what we have added to our platform, it's not entirely being used right now because it's still new, but it's a sharing opportunity. So if you want to use the data from a Danish salmon, a big Danish RAS facility, you will have to ask them and you will have to tell them what you want to use the data for. And you will have to argue that this is something that actually benefits fish farming. And this is like our approach to do it. If everyone is gathering the data for themselves, like many people do, many companies do, I think it would be impossible to use it for the benefit of the sector.
I mean, here I think we can learn a lot also from the from non-blue economy developments, because the data sharing and use of data is a huge discussion when it comes to energy data, for example, or just resource efficiency in cities. So regarding all of these IoT solutions, I think they're a bit underdeveloped in the blue economy sector. And for sure, there should be some good examples and good opportunities to learn from very other sectors that are not represented here today. How maybe Mette or also Per and Anna, how are you approaching the bigger data platforms on the EU level, like IMODNet, for example, or we have Copernicus that are more collecting satellite data. Are those platforms something that you are actively engaged in, trying to contribute to sharing data?
We don't do that, not yet, but we intend to do it eventually, but we don't do it now, no.
Yeah, I guess also for you, if it's the farmer that owns the data, it's the farmer's decision if it should be shared to any other platform as well.
Yeah, but we are also adding data to the platform for them to use. Like we intend to do it. Like for our sea cage customers, we would like to add data on current, water temperature, all that geological and meteorological data, and we might use third-party data for that.
Per?
I think what we're doing, it's more or less kitchen table technology, because the aim is that to develop technology that can be used by the mussel farmer, or so we do not need huge computers. So our focus is much more simple. The YOLO programming structure is also really limited. I mean, so we are not dependent on really heavy computers to calculate, but I mean, we can use standard software and standard computers.
Does anyone from the audience have any questions for the speakers or for each other? We also have the chat if anyone wants to write something. No. Good. I don't have any more questions to any of the speakers. And if no one else does, I think maybe we can make it a shorter meeting and get some more precious time. And yeah, thank you everybody that have been here today. It's been very, very interesting. I'm happy that it's been a nice engagement, and a big thank you for all of the speakers. Before we leave, I would like to get some input both on the new format of the working group meetings. Before, we've kind of separated everything into the different working groups, but we've realized that there are a lot of cooperation possibilities between the different value chains. So this year we're going to continue with this format of joining them all together and making it a bit longer. And if you have any input on the new structure or the new format, please let us know. But this has enabled us to also plan a bit further ahead, and I can already now tell you that the next working group meeting is going to be 17th of April. And that time we're going to discuss sidestream uses. So again, it's going to be a joint working group meeting with all of the working groups. And it's going to be more on the topic of circular economy. And I think we're going to be able to highlight even more exchanges between the different value chains than today. If you have input on speakers, or if you would like to share anything from any of your products or from your organizations, maybe companies, then I will send out a link to a form so you can submit a suggestion. And with that, I think that we can close the meeting. And again, I want to remind you to keep updated on Blue Biomatch. That's also where you registered for the webinar. All of the updates for further meetings are there, and you can also connect with a lot of people working in the sector. It would be great if you could keep the discussions there, post in the forums, and look for collaborations. And with that, I thank you all for being here, and have a nice rest of the day. And especially thank you to all of the speakers for great presentations.
Thank you.
Thank you.
Thank you. Bye.
Thanks so much.
Thank you.
Bye.
Thanks a lot. Thank you. Bye.
Thank you.